Alright, 1 second. Sorry. I overflew it. I overflooded. God, I can't even talk. I overflowed a tank right before I started the stream. Wait 1 second. Keep each other company in the chat. Give me 1 second. But I see you guys. Just gotta clean this up. Holy. No one noticed I said over flooded, right? Like, we can just forget that happened. Cool. JP didn't notice anything. What's up in the flesh? What's up, 186? What's up, Big Steve? Adam, how's it going? Oh, Diana in the building. Holy. All right, 3 more minutes. Big Steve said, Nick is the guy that calls you and says, hold on. What do you guys think of this macro shot? Should we keep it or should I change it? Let me know. Okay. People coming in. Sorry about that. I was just cleaning up a spill. Thanks. Thanks for the towels. All right. I'll get my mic plugged in. Listen, you guys want to stream? This is what it's going to take. This is what's going to take. Research paper out to fill up the screen anyway. I'm going to try and... I usually try and leave some space so you can... By the way, if you guys think I can make the space of the things I'm reading smaller so you can see the macro shot more, let me know. Because, I mean, if you guys aren't reading that shit, then I don't need it to be on the screen there for me. All right. Audio source. Okay, we are official. What's up? How's it going, Johnny? Johnny said, I like the shot, especially when the shrimp was on it. I just thought it was an interesting composition because, like, the rock is in the middle and it goes out of focus kind of, kind of across the whole screen. It goes in and out of focus, so you get some interesting stuff. Steve said, where's Mickham? I want to ask him about these Antuta. Yeah, where's Mickham? And the flesh says change. Good for now, I guess. Oh, my God. Oh my God. What's up? What's up in the flesh? How's it going? What's everyone up to? I got some good... Listen. when the MVPs make a request, I gotta come with the stream topic quickly. So SVC's been asking about genetics, a genetic stream for a while. It was pretty hard. Well, no, it wasn't hard to find something, but I didn't want it some blind leading the blind situation. We already do enough of that here. Let's be honest, okay? I mean, at least I'm honest about it. I don't want Glassbox to make a video about me talk about, look at this talking head. He live streams for 3 hours spreading lies. You know what I mean? So, but I think I found some stuff that I can read. Now, after the last stream, I had to take the research a bit more seriously. I don't know. I was so annoyed at how horribly I was reading and comprehending that thing. So while I was there's some downtime sometimes at work. So I read the parts of this book. I would say I read 70% of it, but I stopped when it was talking about things I had no interest in. I stopped reading. But I picked up this book at the library, Painless Chemistry. I was like, I gotta start getting more familiar with some of these concepts. I probably need to pick it up again to absorb the information a bit more, but I was like, okay, let's work on it. So it's kind of the same thing with just this genetic stuff. I'm sure after the stream, I'm gonna be like, oh my god, I gotta pick up a book, and then we'll be back at it again. I'm gonna change the camera angle, and then let's get into it. You know what I'm saying? Let's get to rating. SVC said, really ready to learn about genetics. Robocop is life. Or it said JP. Giants are very cool of the plant next to a rock. Ram's horse, nice mix of colors and textures. Yeah, let me change it up in a second. What do you guys want to start with? There's this one paper. I kind of had an idea where to start with, the basic aquaculture genetics paper, because it defines a bunch of stuff in simple words. And I figured that would be a good place for us to get a start. There's a couple of really cool beta fish papers. That's one of the easiest ones to find good stuff on, including one where they edited the genes with CRISPR on it. So if you have a preference in order, let me know. I think I'm going to start with the aquaculture genetics one, though. Move this around. Get the camera a little bit closer. It is a macro lens after all, so let's make use of it. Okay, maybe that's a little too close. Hold up. All right. So, as I was saying, let's see if we can get into this some. I just gotta decide on my camera angle. I'm gonna move it one more time. I'm sorry. I can't help it. I'm going with that. All right. Adam says, where do you guys find these papers? I mean, you can find them all over. It's pretty, just Google. Really, is primarily what I do. I primarily just Google things. Now that I've killed the stream with 3 billion hours of downtime, let's start on one of these. So, and like I said, let me know if there's ever a situation where you feel like you don't need the words on the screen to be that large. Because sometimes I wonder how much I really need what I'm reading to be shown. How many of you actually read what I have up on the screen? You know what I mean? That's what I wonder sometimes. So, let's get this up. 186 said, Barron's reminds me of school. I know. I felt like I needed to go back in time. I basically remember none of this stuff, man. I'll be honest. It was bad. But we're working through it. So, getting this on the screen. There we go. Yeah, I like having as much of the background as possible. Maybe I can go with like the whole left side of the screen being open kind of a thing. Like this. No, maybe I like this. Or maybe this is still better. It don't matter. All right. I thought that this was nice. This is from January 2010. The Southern Regional Aquaculture Center. What I liked about this one, though, is I felt like, we care more about the ornamental fish stuff, obviously, but I felt like it defined enough concepts and things like that that was kind of helpful for just understanding as we move forward. I tried to make a path. Chickens that I do genetics are important. I like paying attention. I want to people I want to hear talk. Okay. JP said this is great. Need to watch some blue bolts. Yeah. I got the cup to pour the lemonade in. I poured some in, but I just took a sip of lemonade, then picked up the can and took another sip of lemonade. Doesn't make any sense. All right. Well, yeah, there are some blue bolts in there. The blue shrimp on the screen, though, are some ocean blue shrimp that I got from Just One More Fish. I think they're pretty nice. The babies are starting to grow. There's only been one or two that I've taken out. I'm like, I'm going to cull you. But otherwise, most of them, they got nice blue legs and all the good stuff that you're looking for in a shrimp. So, let's get into it. The basic aquaculture genetics. All right. By Jason W. Abernathy, Eric Peatman, and Zhiheng Liu. That's how I'm going to say it. All right. The inheritance of desirable traits in crops and livestock has been the foundation for selective breeding for thousands of years. However, our understanding of genetics, which is the biological basics of heredity and variation among organisms, has deepened dramatically in the last several decades. This knowledge expands the potential applications of genetics and genome technologies in agricultural practices and aquaculture settings. Current genetic improvement programs focus on selecting superior brood stocks, using better breeding practices, increasing sustainability, and minimizing environmental problems. These programs have already led to more efficient, productive, and profitable aquaculture systems, but the genomics revolution promises to speed and amplify genetic advances in the near future. This publication describes both basic genetic principles. See, that's why I picked this one, because that's what we need. Basic genetic principles and applied genome technologies that are relevant to today's aquaculture industry. So I feel like with that introduction, we should all be properly excited for this. Introduction to genetics. Genes and chromosomes. The basic unit of inheritance is the gene. Genes contain the biological code for the production of observable traits or phenotypes in an organism. Genes are arranged on a large molecule called, this is our first thing I'm going to look up, this word, how to say it. It's spelled DEOXYRIBONUCLEIC. Okay, I'm listening to the thing. I can't read this phonetic stuff. 1 second. Let's see what the audio says.
It's confusing.
Whoa.
So make sure to stay tuned to the channel. So how do you say DNA in full? Deoxyribonucleic acid. ribonucleic
Holy crap.
deoxyribonucleic acid.
Deoxyribonucleic acid. Okay. Well, hope I never have to say that again. All right. Deoxyribonucleic acid, DNA. Seems like one of those things I should have just been able to read, right? DNA consists of subunits called nucleotides. The 4 nitrogenous bases that define the 4 different nucleotides are adenine, guanine, thymine, and cytosine. We have A, G, T, and C representing those. Diana said mouse, Mickey Mouse, Hickey Mouse. What's going on here? Got nursery rhymes. It's the combination of these subunits in a linear arrangement that codes for a gene. Most of the DNA forms a structure known as a chromosome. The chromosomes are located in all the nucleated cells. The total number of chromosomes can differ among species, but will generally be constant in the same species. Most aquatic species contain 2 sets of chromosomes, one inherited from the father and one inherited from the mother. Any organism with 2 sets of chromosomes is termed a diploid, or 2N. We will focus on diploids in our explanation unless otherwise noted. The karyotype is the general appearance, size, number, shape of an organism's chromosomes. There are 2 types of chromosomes in the eukaryote. Oh my god, bro. I'm already sweating.
Eukaryotic.
Eukaryotic. Okay, and eukaryotic species with nucleated cells. Autosomes and sex chromosomes. Autosomes are all chromosomes that are not sex chromosomes. Sex chromosomes usually determine the fish's sex, and we have a nice little diagram here. Deoxyribonucleic acid. Here we go, right here, and then we have chromosomes. And it says Figure 1: the physical structure of the deoxyribonucleic acid DNA characterized as having the double helix structure with a sugar phosphate backbone. The nucleotides pair with each other to form the structure. Adenine pairs with thymine, with guanine pairs with cytosine, and this is from the National Human Genome Research Institute. That's another thing I liked about this one. It had some pictures. Oh my gosh, what is SVC doing to me? Okay. Autosomes are usually designated with a number, while the characteristic notation for sex chromosomes is XX and XY in diploids, where XX are females and XY are males in the XY sex determination system. Many species of fish can have different sex determining system. Some may only contain autosomes, some are hermaphroditic, and either change sex as they grow or, in rare cases, possess both male and female organs simultaneously. In some fish species, the sex chromosomes have not yet been determined. I'm going to have to listen to how to say this again. Bro, how do I forget?
Eukaryotic.
Eukaryotic. Oh my gosh. Even though most eukaryotic species are diploid, there are exceptions to a number of chromosome pairs in some aquatic species. Further, the number of chromosome sets can be manipulated in genetic improvement programs. Haploids, parentheses N, can be created, as well as fish that contain chromosomes from the mother only, gynogens, or from the father only, androgens. Triploids have 3 sets of chromosomes, that's 3N, and tetraploids have 4 sets of chromosomes, 4N. In nature, tetraploids are more common than triploids, and the channel catfish, oh my gosh, I just accidentally skipped ahead. Oh, 1 second. The channel catfish is a diploid and contains 29 pairs of chromosomes. So that's 2N, so diploid, 2N equals 58. The salmonoids, salmon and trout, and the common carp are widely thought to be tetraploid, 4N. What's up, Uncle Smiley? How's it going? Since chromosomes occur in pairs in diploid organisms, each gene has at least 2 copies. Each copy of a gene called allele, A-L-L-E-L-E, is located at a specific location on each of the sister chromosomes. This location is called the locus of the allele. I'm going to look up how we hear this being said, too. JP says, how zoomed do you have to be to see the microscope to the DNA ladder things? That's a good question.
Allele.
Allele. Okay, allele. I remember this. 2 alleles can be identical or have variations in their DNA sequence. If alleles at a specific locus are identical, the organism is homozygous for that gene. Conversely, if alleles at specific locus are different, then the organism is heterozygous for that gene. Differences in alleles within an individual can produce genetic variants and thus different phenotypes in a population. This stuff is definitely going to come up later, so hopefully you guys are paying attention when I forget, you can remind me. The combination of alleles for a given trait is called the genotype. Chromosome replication and cell division. There are two forms of cell divisions, mitosis and meiosis. As an organism grows, cells must divide and replicate to increase in number and to replace older dying cells. Mitosis is the process by which a somatic cell divides to produce two identical daughter cells. Somatic cells are all cells except egg and sperm and the cells that produce them. During mitosis, the entire cell is duplicated, including the chromosomes and all other cellular material. Thus, each daughter cell contains a diploid set of chromosomes. This process differs from meiosis, which is associated with a gamete, egg and sperm formation. The eggs and sperm undergo cell division and replication, but produce cells that contain a haploid gamete, containing only one set of each chromosome pair. The process of creating haploid gametes is critical to reproduction in diploid organisms. When an egg is fertilized by the sperm, the correct number of chromosomes, 2N, is recovered in the zygote. The process of sperm or egg generation is called gametogenesis. More specifically, the production of sperm cells is referred to as spermatogenesis, while the production of egg cells is called oogenesis. In meiosis, at least three important processes occur to produce genetic variability in the sperm and egg. By the way, here's a little diagram up here, the basic principle of meiosis, also from the National Institute of General Medical Sciences. So up here, we have the chromosomes from the parents and the cell nucleus. The chromosomes replicate. The like chromosomes pair up. The chromosomes swap sections of DNA. Nucleus divides into daughter nuclei. Chromosome pairs divide over here. Daughter nuclei divide again. Chromosomes divide. Daughter nuclei have single chromosomes and add new mix of genetic material. So, nice little diagram there. So, in meiosis, at least three important processes occur to produce genetic variability in the sperm and egg: crossing over, segregation, and independent assortment. First, the chromosomes and the gametocyte. I'm going to look up how to say this too. Oh my gosh. I just want to be sure. You know what I'm saying? I love how all the pronunciation videos, like, they make them like at least a minute long. They don't just come out, they're like, yeah, let me just say this word. They're like, so here I'm going to teach you content farming.
Gametocyte.
Gametocyte. Oh yeah, okay. The gametocyte must undergo replication. After chromosome pairs replicate into diploids, the homologous chromosomes form pairs and bundles of four called tetrads. At this point, the chromosomes that form tetrads can wrap around each other. The chromosomes may then break apart, and pieces from different chromosomes can rejoin. Therefore, genetic material has been swapped from one chromosome to another. A portion of a chromosome that originated from the mother is exchanged between the homologous chromosome from the father and vice versa. This process of recombination or physical exchange between homologous chromosomes is called crossing over. This occurs in the cell stage known as the primary gametocyte, and the other processes that increase genetic variation include segregation and independent assortment. After chromosome duplication and crossing over in primary... One second. I want to listen to it being said again. Holy.
Gametocyte. Gametocyte.
Gametocyte. Oh my gosh. Over the primary gametocyte, the chromosome complement must be reduced from the diploid state to the haploid state, and a cell division step for both the egg and sperm. Each chromosome pair will separate, going from a primary gametocyte to the secondary gametocyte. Each secondary gametocyte will contain a single replicated chromosome from each homologous pair. In a spermatogenesis, all chromosome pairs separate, and one of the chromosomes of each pair goes to a secondary spermatocyte. In oogenesis, all chromosome pairs separate, and one of the chromosomes of each pair goes to a secondary oocyte or polar body. The process by which the duplicated chromosomes separate and form secondary gametocytes is the basis for Mendel's law of segregation, which I expect you all to be familiar with, as I obviously am. But if you're not, here's a little figure. Figure 3. Mendel's law of segregation. This is a classic example of independent segregation of alleles using a single observable trait. Gregor Mendel used the observable trait of seed coloration in his plant experiments. In this experiment, the dominant allele is yellow seed color, which is A. You can see that up here in the diagram, which is A, whereas the recessive allele is the green seed color, which is the lowercase a. So I guess we got uppercase for dominant, lowercase for recessive. One homozygous dominant, that's AA, and one homozygous recessive, the lowercase aa, up here. So those one homozygous dominant and one homozygous recessive parent were crossed to produce F1 progeny that are all heterozygous dominant, which is uppercase A and lowercase a for the trait, and all yellow seeds. A cross between the F1 progeny, so the uppercase A, lowercase a, cross with another of the same set, produces F2 progeny with 3 genotypes. 2 big A's, 1 big A, 1 small a, and 2 small a's. And 2 phenotypes, yellow or green seeds. Seed coloration is characterized as homozygous dominant, 2 big A's, yellow heterozygous dominant, 1 big A, 1 small egg, yellow or homozygous recessive, 2 small a's, which is green. All right, you guys following all that? Holy, what's up, Cam Jam? How's it going? Beto, how's it going, Beto? Hope work's going all right. Now, I'm not gonna lie. So far, this is what I've understood the best, and I feel like this here is probably pretty relevant for fish breeding stuff. So yeah, this will be on the fabled test for sure. Mendel's law of segregation. This is definitely gonna come up again. Mendel's law of independent assortment states that each gamete receives a random mixture of alleles, one at each locus from each parent. The process of segregation and independent assortment are random in that each secondary gametocyte or polar body receives a random mixture of chromosomes from the mother and the father. The reduction of chromosomes from the primary gametocyte to the secondary gametocyte reduces the number of chromosomes for the diploid complement to the haploid complement. This process ensures that when a sperm N fertilizes an egg N, the correct complement of chromosomes 2N will be created in the zygote, the fertilized egg. The exception to the independent assortment of chromosomes and alleles is linkage. But before we get to that, let's look at Mendel's law of independent assortment of alleles of different genes. We have another figure. JP says, you know it works with peas. Okay. So, he also observed the trait producing seed shape, either smooth or rough seeds, along with seed color. Goodness. Let me make this a little bit longer for you guys. There we go. Along with seed color. In this example, the dominant alleles are yellow, big A, and smooth, big B seeds, whereas the recessive alleles are the lowercase a and the lowercase b seeds. Sorry, the recessive alleles are green. Green is the lowercase a, and rough, lowercase b seeds. So we have that up here. The parental cross produces F1 progeny that are all heterozygous dominant, big A, small b, big B, small b for the traits, and thus yellow smooth seeds. The F2 generation produced by a cross of F1 generation, big A, big B, small b, crossed with the same, yielded progeny with various genotypes and phenotypes. 2 new phenotypes appear in the F2 generation: yellow rough seeds and green smooth seeds. Since the genes were segregating and assorting independently of each other during meiosis, multiple combinations of alleles and traits were produced. Note that independent assortment of genes may not hold true for all combinations of alleles, such as linked with genes. All right. So linkage. The genes that control a trait are located on the same chromosome and are close together. They're said to be physically linked. In this case, these genes may be inherited together. The secondary gametocyte consists of two secondary spermatocytes, males, or secondary oocytes in polar body, females. Each secondary oocyte normally produces only one haploid egg. The rest are polar bodies. In females, after segregation and independent assortment, chromosomes at one end are pinched off with a little surrounding cytoplasm. This forms the first polar body. Polar body cells are non-functional and are a byproduct of oogenesis. Since the egg is the major site of nourishment to the embryo, a high concentration of cytoplasm is necessary, and hence the unequal division of cytoplasm in the oocyte versus the polar body. In the flesh says baking a cake, getting make bake one. Betta says, what's up, Nick, Smiley, KJ, JP, in the flesh aquatics, Jacob and Diana. What's up to all you people? The first polar body may or may not divide again to produce two small haploid cells. The other daughter cell is the secondary oocyte. The secondary oocyte in the first polar body produce one mature egg and one secondary polar body. Secondary oocytes are not stimulated to egg production until after they have contacted the sperm. In males, the two secondary spermatocytes produce 4 sperm cells. All sperm cells have the haploid number of chromosomes that have been independently assorted and contain equal amounts of cytoplasm. By the way, I need to get the link to this in the chat. 1 second. I forgot to do that. Link to what I'm reading in the chat if you want to read for yourself. And I'll put it in the Discord live stream discussion channel as well. Bam. Look at me. Good practices. Okay. You're getting your bacon? Okay, very nice. Get that bacon. Let's go. Okay, where was I? Right. Development. Developers. All right. I'm not even old enough for that. Okay, when an egg and sperm are fused through fertilization, a diploid zygote is produced. This is a single cell. Mitosis of the zygote and subsequent daughter cells is responsible for the growth and development of the fish. Fish eggs are mostly composed of yolky cytoplasm. In general, after fertilization, oh my god, they said fish eggs. Oh, finally a word I understand. Oh, let's go. Sorry. My brain, I was like, wait, I know what they're talking about right now. Let's go. Fish eggs are mostly composed of yolky cytoplasm. In general, after fertilization, cell division begins in a thin, yolk-free region of the egg called a blastodisc. This initial cell division is known as the cleavage phase, holy, and the initial cells are termed blastomeres. Blastomere divisions are rapid and cells build upon one another to constitute blastoderm. It's a Pokemon. These early cells mix and can give rise to a variety of cell and tissue types. After a period of rapid cell cleavage, cell divisions begin to slow and cell movement begins. Blastomeres begin to cluster and segregate through the embryo. After the blastoderm has filled about half the yolk, or sooner depending on the species, germ layers form to begin the process of gastrulation, or cell restructuring. These layers will develop into the tissue and organ systems of the fish. From a genetic standpoint, zygotic gene expression generally begins during the cell division stages. Sex determination. Now remember, we got to get through this stuff so we can actually get to, all the different color. We need to have a foundation here, okay? So hopefully you guys you're sticking with me. Sex genes are the main determinants of gender. The sex chromosomes of different fish species may or may not be morphologically distinct. Many sex chromosomes resemble autosomes, and/or some sex-determining genes may be located on autosomes. In these cases, sex-linked traits must be studied to determine the sex chromosomes or sex genes that are located on autosomes. The most common sex-determining system is the XY diploid system. This system is the most common in known fish species, as well as in humans. Channel catfish have the XY system. The system was so named because the sex chromosomes in human resemble a Y or an X. In the XY system, the sex chromosomes in females are identical, XX, while those in males are a mix, XY. The Y chromosome is only found in males. Since the pairs in females are the same, these chromosomes are termed homogametic. Homogametic. You know what? They've been saying these words in ways I would never imagine. All right, how to say it. Let me just listen to this thing again. Holy. Emma Sane. Shout out to Emma Sane, bro. She be saying everything. I wonder if when they were growing up, their parents were just like, yo, you never shut up. You say every word in the dictionary.
Homogametic.
Homogametic. Homogametic.
Homogametic.
Homogametic.
Homogametic.
All right, I got it. Homogametic. Oh, yes, perfect. While in the male, they're called heterogametic. All eggs contain only a single X chromosome. In males, half the sperm population contains a single X chromosome and half contains a single Y chromosome. In the XY system, the heterogametic male is the one responsible for sex determination in the population. If an egg X is fertilized with a sperm carrying the X chromosome, the sex of the offspring will be female, XX. If an egg X is fertilized with a sperm carrying the Y chromosome, the offspring will be male, XY. Again, the resulting offspring will be diploid. It is likely that the only portion of each sex chromosome is ultimately responsible for sex determination. Other traits, genes, may be located on the XY chromosomes in many different aquaculture species. There are several other sex-determining systems in fish besides the XY system. Oh god, one is the WZ system. Nah man, I'm Team XY, Team XY. It works exactly the opposite to the XY system. A homogametic fish is a male, while a heterogametic fish is a female. Blue tilapia is thought to have the WZ sex-determining system. The female blue tilapia contains a different set of sex chromosomes, WZ, while the male tilapia contains the same set of sex chromosomes, ZZ. Hence, the W chromosome is the sex-determining chromosome. The sperm, Z, that fertilizes an egg of one chromosome type, Z, will produce males, ZZ, while sperm, Z, that fertilizes an egg of another chromosome type, W, will produce females, WZ. In other words, sex is controlled by different types of eggs, not different types of sperm, as in the XY sex determination system. Another system is the ZO sex determining system. The dwarf gourami fish and a soulfish have been identified as having the ZO system. Here the females are ZO and the males are ZZ. In these species, the females are the sex-determining species and are heterogametic. The female produces haploid eggs with either the Z chromosome or no sex chromosome at all. What's up, Toonie Loon? Camden said, I just hope you set this stream to 18 plus. I always set my stream to not for kids. The females are the sex-determining species and are the heterogametic sex. The female produces haploid eggs with either the Z chromosome or no sex chromosome at all. If an egg with the Z chromosome, Z, is fertilized by a sperm, Z, then the offspring will be male, ZZ. ZZZ. If an egg has no sex chromosome, O, the resultant fertilization will produce a female offspring, ZO. The exosystem also exists in some species of fish, including a sunfish species. In this system, the males are heterogametic, XO, and determine the sex of the offspring. In a way, the XO sex determination system is similar to the XY sex determination system, except that in the XO system, the males contain just one set of sex chromosomes. What's up, Yuri? How's it going? Several other sex determining systems have been discovered in fish. Many of these systems are more complex than the XY system and its variants. These systems have multiple sex chromosomes. One example of a multiplex system is the WXY sex determining system. Now we're getting into radio stations. Common to the platyfish used in aquaria. Here, both males and females can either be homogametic or heterogametic and determine the sex of the offspring. In WXY, the W chromosome acts as the modifying chromosome that can block the male determining ability of the Y chromosome. Therefore, the XY and YY offspring are males, and the XX, WX, and WY offspring are females. Other multiplex systems include the, oh my gosh, this is X1, X1, X2, X2 slash X1, X2, Y, or ZZ slash ZW1, W2, we're doing taxes now, and XY1, Y2 slash XX systems. My brain hurts already, bro. I'm dying. We're getting somewhere. Finally, sex-determining genes also can be located on autosomes. Some aquaculture species do not have any morphologically distinct sex chromosomes. In those species, sex is determined by a number of female or male genes located on specific autosomes. Later, there's so many pictures in the Betta fish studies, man. I'm like, we got to get through the foundation, though. One important concept to introduce is genetic sex versus phenotypic sex. Fish, being lower vertebrates, have much flexibility in sex organ development, so phenotypic sex may not reflect the chromosome composition of the fish. A number of factors can influence expression of sex phenotypes. Some of the most common factors are temperature, salinity, population density in tanks, pressure, hormone treatments, radiation, and photoperiods. My goodness. Yo, imagine if like you end up a girl because your mom left the lights on too long. It's getting crazy. Sex modifications may be induced or may occur naturally in some populations. Yuri said, I gotta go to sleep. Take it easy. Thanks for stopping by. In the Flesh sounds like a cheat code for a video game. For instance, hormone treatment, estrogen, of developing males in some fish species can produce phenotypic adult females even though the fish contain XY chromosomes. Conversely, hormonal treatment can lead to the development of all males even though the fish have female genetic makeup. For instance, a common practice in aquaculture is treating tilapia with 17A methyltestosterone, MT, to induce an all-male population, as male tilapia grow much faster than females. Such hormonal treatments leads to increased yields. In such cases, however, the fish may harbor XY chromosomes, or they may harbor XX chromosomes. Hey, yo, don't let the homophobes know they're eating transgender fish. Now, this is getting crazy. Yo, what in the world? Oh, sorry. Science is so funny. It's so funny. 17A methyltestosterone. Okay, so what did they say? Phenotypic sex versus genetic sex. Got it. Qualitative genetic traits. Phenotypes can fall under the category of qualitative traits, or traits that can be simply described as one or the other. A trait is qualitative if it can be sorted into one of at least two categories. These traits are not measured over a range, as is the case with quantitative traits. An example of a qualitative trait would be fish pigmentation. So fish pigmentation, that would be a qualitative trait. The fish is either normally pigmented or albino. Qualitative traits are often the simplest to characterize because they're likely to be controlled by only one or a few genes, unlike quantitative traits. Since aquaculture species can be categorized by these traits, the population can be described by the ratio of its members with these traits. The number of fish with each trait are simply added up and described such as 3-to-1 or 1-to-1 and so on. JP says locked in, let's go. For example, a population of 20 fish has either a fan tail or a round tail, a qualitative trait. if 15 fish have fantails and 5 fish have roundtails, the ratio of fantails to roundtails is 3-to-1. Described another way, 1/4 of the fish population has roundtails and 3/4 of the population has fantails. Qualitative phenotypes are the same in different environments. A number of factors influence these ratios, including the number of genes needed to produce the phenotype in gene action. The genetic mechanisms described here are typically referred to as classical genetics or Mendelian genetics. Gene action can be characterized as a single or multiple genes producing a qualitative phenotype. Classic qualitative traits are dominant or recessive. If a single allele is expressed over the other at the same locus, then the mode of action is termed dominance. Complete dominance mode of action describes the expression of alleles at the same locus, where one copy masks the effect of the other copy. The phenotype expressed is termed dominant phenotype, and the other is the recessive phenotype. When dominant alleles occur, there are 3 genotypes possible, while only 2 phenotypes can be produced. A classic example of complete dominance of a single locus is the gene for albinism in the channel catfish. Now we're back to the channel catfish. The dominant genotype that produces normally pigmented catfish can be labeled as AA, so 2 big A's, or 1 big A and 1 lower A, where the capital A is the dominant allele and the lowercase a is the recessive allele. Both the capital double AA and the capital A lowercase a fish produce normal pigmentation. Only fish with the complete recessive pigment gene, 2 lowercase a's, will be albino. Hence, with dominance there are 3 types of genotypes. With dominance there are 3 combinations of genotypes. 2 capital A's, 1 capital A, 1 lowercase a, and 2 lowercase a's. While only phenotypes are produced, the normal coloration being the 2 capital A's or the big capital A lowercase a. Fathom says, I take a break from my biology homework just to hear Nick talk about biology. Oh my god, bro. Can you help us, Fathom? Can you help us? We're at school too. While only phenotypes are produced, how you doing, by the way? The normal coloration has 2 uppercase A's or 1 uppercase A, 1 lowercase a, or albino, which is 2 lowercase a's coloration. Fish that are homozygous, dominant, 2 uppercase A's, will obviously be normally pigmented, but so will fish with 1 dominant A and 1 recessive A. With complete dominance, only parents that carry recessive alleles, either uppercase A with a lowercase a or 2 lowercase a's, can produce offspring with a recessive trait. Holy. There are 2 other possibilities concerning qualitative traits, or those are like coloration, things like that. Multiple alleles and sex-linked alleles. Fathom said, are you doing Punnett squares in here? I don't think we are yet. Right now, the paper we're reading is the basics of aquaculture genetics. Diana said, I actually understand this part. I know, the further we go, the more I'm able to understand what we're reading. So I told you guys, if we make it through, I've lined up the papers after this where they should make more sense since we read this first. But, we got to get through it. When alleles that more than one locus control a qualitative phenotype, there are 2 possibilities of gene action. There are epistatic effects or non-epistatic effects. Epistatic effects are interactions between genes that can cause modifications or suppressions of phenotypes. Thus, combinations of multiple alleles at different loci can cause different phenotypes in the simple case of either-or patterns. A classic example of epistasis for a qualitative trait in aquaculture is the scale patterns in the common carp. Common carp scaling includes wild-scaled, mirror, linear, or leather types. Wild-scaled carp have scales all about the fish. Mirror carp have scales scattered around the fish. Linear carp have scales arranged in a linear array. Leather carp have very few scales. These patterns are controlled by genes S and N from two loci. One loci, S, big S, determines the degree of scales, either wild or scaled. The degree of scales. Either wild scaled, SS, or S small s, or mirrored scaled, 2 small s's. The other loci N modify these phenotypes in the following manner. Alright. Let me get to this figure. Alright. The different types. So we got the wild scaled carp, the linear carp, the mirror carp, and the leather carp. All these sound very unappetizing. I'm not gonna lie. Quantitative traits are phenotypes that have a range of expression. These are traits that can be measured rather than either-or traits such as normal pigmentation versus albinism. Phenotypes that can be observed and measured may vary in a population. Therefore, genetic programs must be able to properly analyze and understand the traits of interest. Commercially imported traits in aquaculture include length, weight, growth rate, feed conversion, oxygen tolerance, percentage of body fat, meat production, disease resistance, and stress resistance, just to name a few. These traits are said to be quantitative because they typically vary among individuals within a population. Quantitative traits are measured using a continuous distribution system and statistics. Betta said, I had a magic carp once. Oh my god, he's talking about some Pokemon. Quantitative traits are measured using continuous distribution systems and statistics. Such traits are described and reported around their central tendencies, such as average variance, standard deviation, and range. These traits can be controlled by a single gene, but are usually controlled by several to many genes. These traits are also influenced by the environment. Gene expression levels, the environment, and the interaction of the two can play a significant role in the variation of quantitative traits. Quantitative traits are under a constant variance. Each gene, except linked genes, is segregated and independently assorted. As many quantitative traits are controlled by multiple genes, each of those genes will be segregating and sorting independently as well. Since so many genes are involved and each locus is independently assorted, the potential for a variety of genetic combinations in the offspring is large. Because quantitative traits are also influenced by the environment, the action of both the environment and multiplex of genes involved produces the distribution of these traits in a population. So it kind of sounds like the quantitative genes are a pain to determine which is causing which, environment or the genes. As we've seen in some of the papers, like the cavefish, the blind cavefish paper, when it was talking about the growth rates. That comes to mind. Genetic variance and breeding. Genetic variance is a key component in exploiting the traits of interest in a breeding program. From the equation above, components of genetic variance include epistatic effects, additive effects, and dominance effects. Epistatic effects are rarely used in a breeding program because there's so many possible combinations of alleles in this measurement of genetic variance. Most programs focus on additive and dominance genetic variance. The relative contribution of either of these effects on the phenotype of interest determines the best approach to use in a breeding program. Additive effects are passed down from parents to offspring, and dominance effects are created with each generation. If additive effects are large, and thus the variation within a population is large, fish with a desirable trait can be selected and bred. If the additive genetic variation is small, then selecting fish for a specific trait within a population may not produce progeny that express the trait better than the parents. In this case, a recombination of alleles would be the expected approach in the breeding program. More alleles are introduced through an influx of new genetic material based on trial and error. New strains of fish can be introduced into the breeding program with interspecific hybridization, or different species bred interspecific hybridization. So intraspecific hybridization versus different species bred interspecific hybridization. Intra and inter. Okay. A new combination of alleles may produce offspring that harbor the desired combination of phenotypes. For a selection program to be effective, the amount of additive variance should be determined. The measure of heritability, H2, describes the proportion that additive genetic variance contributes to a phenotype in a population. Heritability describes the percentage of a phenotype that can be inherited in a predicted manner. This value should be reliable, since for a quantitative phenotype, the genotype is not affected by meiosis. These value ranges from 0 to 1 and are a percentage. A heritability value of 1 suggests that the phenotype observed is 100% explained by the additive genetic variance. A heritability of 0 suggests that the additive genetic effects do not contribute to the phenotype. In general, a heritability value greater than 0.2, H2 equals 0.2, suggests that the trait may be reliably exploited in a selection program. If a trait's heritability is lower than H2 equals 0.15, a selection-based program may not be the answer because dominant genetic variance is more important in this case. Just remember that we're dealing with quantitative traits here. These traits may be affected by the environment in the population and can vary between generations. Direct selection of traits with low inheritance may be difficult. In such situation, genome-based technologies such as marker-assisted selection may be more applicable. Now we're getting into the stuff that actually is relevant to, breeding some fish or something. Genetic improvement programs. Selection. Selection is a process in which individuals with desired phenotypes for a particular trait are identified and used as future broodstock to produce progeny that are also superior for the trait. Selection programs may include mass selection and family selection. In mass selection, the performance of all individuals is compared, and selection is based on the performance of each, disregarding the parentage. In family selection, the average performance of families is compared, and whole families are selected. Selection programs are reliable only if the genes responsible for the genetic variation are passed to the offspring. Additive genetic variance is transmitted to the offspring in a calculated and reliable manner. Heritability, H2, values must be taken into account. These values are a direct measurement of genetic variance explained by additive genetic effects. The heritability estimates for a trait should be as large as possible to ensure that the efficacy of a selection program. Many traits may be correlated positively or negatively. For instance, fast growth is often correlated with a more efficient feed conversion rate in some species, and the opposite can be true in other cases. Therefore, selection for fast-growing fish also selects for fish with better feed conversion rates. However, if the traits are negatively correlated, the breeder must be careful because a selection for one trait may negatively affect the other. For instance, fast growth may be negatively correlated with reproduction capacity, so that selecting for fast-growing fish could reduce reproduction capacity. Therefore, in selection programs for certain traits, other important traits must be carefully monitored to determine if there's any correlation among the traits. I know I was talking to this one guy who was breeding a strain of something about black mollies, and he said whatever gene responsible for the super black color also was terminal. They did not live very long. So, yeah, you got to be careful. Hybridization. If heritability is very low, selection methods may not be the best way to increase performance. To improve a trait in this instant, greater genetic variation for new combination of alleles is needed. New combinations can be created by mating fish with different genetic histories, a process called hybridization. All offsprings are called hybrids. The parents can be of the same species but different strains, so that would be intra-specific hybridizations. Or the parents can be of different species, interspecific hybridization. Improving a trait using hybridization is done through trial and error. Some of the hybrids may have superior traits and some may not. But hybridization is the best way to increase performance when the heritability for a trait is very low, because hybridization creates new combinations of alleles through dominant genetic variance. This process is independent of heritability, so hybridization programs can be used even when heritability is high. Selection hybridization can both be used to increase fish performance. Cam Jam's going to bed. Good night to Cam Jam. Sleep well. Interspecific hybridization can be used to improve fish performance in one of two ways. It can be used to produce a new strain that can undergo selection for a trait, and it can also be terminal, with the hybrids being the end product. In general, hybrids have better fitness because of greater genetic variability. This is known as hybrid vigor, or enhancement through outbreeding to create a new breed. A cross must be made between two individuals of two strains with different genetic backgrounds, followed by a selection program to improve performance. A selection program using hybrids usually cannot begin until the second generation of fish is spawned. This is largely due to the principle of dominance genetic variation. First generation hybrids generally do not pass on the hybrid vigor, that's important to remember, and the superior traits to all offspring. I'll say that one more time. Haha. First generation hybrids generally do not pass on hybrid vigor and superior traits to all offspring. The hybrids are created using two distinct strains, and therefore additive genetic effects should be increased. This means that the subsequent generations are suitable for use in a selection program. What's up, Lost Stephen America? How's it going? Another approach is to conduct a selection program within a strain and then use hybridization to try and improve performance. However, not all hybrids will show improved performance. Success not only depends on the strain selected, but also on the reciprocal hybridization. This means that a male of one strain crossed with a female of a different strain may produce progeny with different traits and vice versa. The hybrids that perform the best will be determined by experimentation. And then we have interspecific hybridization. So that was intra, now we're at interspecific hybridization. Fish of different species may be crossed to produce more productive progenies. Interspecific hybridization is usually used to exploit hybrid vigor, or the tendency of a crossbred organism to have qualities superior to those of either parents. However, if progenies are fertile, interspecific hybridization has also been used to improve genetics through introgression, a process by which the genes of one species flow into the gene pool of another. This is achieved by backcrossing an interspecific hybrid with one of its parents. The same principles apply as with intraspecific hybridization. Genetic improvement is based on new combinations of alleles. Interspecific hybrids must be able to produce progeny. Once this is established, a breeding program can be attempted, but many between-species hybrids are sterile, do not reproduce as readily as parents, or produce progenies that are non-viable or abnormal. Spawning does not occur naturally between many species that can be hybridized, so these species must be artificially spawned. The process of producing a superior fish involves experimenting with a combination of strains, species, and reciprocal crosses. An example of successful interspecific hybridization is the crossing the channel catfish and blue catfish. Now, they love this catfish. Each of these species has different superior traits. Channel catfish is best for commercial production because of its growth rate, and blue catfish has a more uniform body shape, yields more fillets, is easier to see, and is more resistant to certain diseases. A cross between a female channel catfish and a male blue catfish produces viable offspring. The hybrids have faster growth rate and are more disease resistant than the parental species. With this knowledge, hybridization programs can produce fish that can be selected for further genetic improvements. All right. If you want to read the rest of this one, you're on your own. I'm going to pause there. I'm going to load up the next one. I think that's most of what I wanted to get into with that. How are you guys feeling? I kind of like starting with the most boring ones first. I'm not going to lie. But I thought that was a good little basis there. The next one has a good amount of photos. The next couple of ones. This is going to be about betta fish. So I think we're going to get to see some of these principles play out a bit more clearly. So let me load this up. But how's everyone feeling? Are we still here? Dwarf Cyclidiot said, "This is why I changed my major from bio to history in college." Hey, man, listen. Like I said, my goal for reading these is just to improve my own knowledge. I don't expect myself to fully understand everything, but I think as we kind of read some of this, some concepts, some vocabulary will start to sink in, and eventually some of the hybrid stuff there, you know, I had heard some of it, but it's good to remember certain things again. JP says pretty good. Flashback to high school biology is fun. Okay, here we go. Betta fish people rise up. This one is the genetic architecture of phenotypic diversity in the betta fish, Betta splendens. Come on. I know this is one that the people in the chat are here for. Where's the betta fish people at? I'm putting the link to this one in the chat, and I'm putting it in the live discussion channel in the Discord. Perfect. George said, I'm glad you did that one, though. W learning. Okay. Hopefully you guys learned something. All right. The betta fish, Betta splendens, is indigenous to central Thailand and the lower Mekong, and is mostly known for its domesticated forms, appreciated as an ornamental fish, but was originally bred for its use in gambling matches, similar to cockfights. Pause. Through captive breeding, a remarkable variety of behaviors and morphologies have emerged, including variation in aggressiveness, pigmentation, body size, and fin shape. Betta splendens is easy to breed and maintain and provides a useful resource for exploration of the genetic basis of behavior and morphology in vertebrates due to the high degree of intra-specific variability. Come on! Look how quickly the last thing we read became relevant. See, you know what? Let's cheers to me. I put this thing together. I put the order of these papers together perfectly. Look at my lesson plan. I mean, you probably understood it more than me, but if you got some water or anything, I'm about to take a sip of my lemonade. 3, 2, 1, cheers. All right. So, due to the high degree of intra-specific variability and the vast number of characterized phenotypes, it also provides a fascinating example of how direct and indirect selection imposed by humans has shaped a domesticated species. Although researchers have examined the inheritance of body color, fin length, and sex determination in classic crosses in the 1930s to 1940s, relatively little was known about the genetic basis of phenotypic variability in betta fish until recent studies investigated the double tail, elephant ear, albino, fin spot, and the SD phenotypes. Here we report a high-quality chromosomal-level genome assembly of a female Betta splendens, resequencing data of 727 domesticated individuals and 59 wild individuals from 6 other species in the Betta splendens complex. We examined the evolutionary relationship and origins among breeds and used association mapping to identify the genetic basis of a number of different traits, including SD, fin morphology, coloration, body size, and aggressiveness and other behaviors. Big Steve said, Petco Bettas, you betcha. Better person in the chat, Diana. What's up, Outside In? How's it going? How you doing? What's up? JP said, I only remember the vocab, very little else. Phenotypic ratios determine traits, so you can kind of predict things. Yeah, I'm right there with you. Going to try breeding a couple pairs of PetSmart Bettas. Very nice. All right. Let's look at this photo here. Let me make my page a little bit wider for this one. There we go. There we go. Hold up. There we go. So, look, here's what we got here. We got B. Samarentalis, Embellus, Stictos, Smardina, Macanensis. Over here we have the domesticated. They got half moon, yellow, white, koi, turquoise, opaque, crown tail, red, dragon, fighter, orange, Dumbo, half moon, royal, copper, veil tail, Dumbo, HMPK, black, and steel. I love the photos. Look at that. We have some different traits down here. So, figure 1. The phylogeny and population structure of Betta splendens. The phylogeny of the telos, including the Betta fish, constructed with single copy genes across the genome. Over here, B is the maximum likelihood tree of concatenated genome-wide single nucleoid polymorphisms, SNPs, for domesticated and wild forms of Betta splendens. The tree is truncated at the branch, connecting to individuals of other Betta species, and the untruncated tree can be found in figure S5. The dashed line circle indicates the wild Betta fish. The letters by the fish photos correspond to the letters in D. The admixture analysis of Betta splendor breeds and their closely related wild species, K equals 12, is presented here, and the results with K varying from 2 to 15 are in figure S7. Acronyms for wild species are as follow. BSP is Betta splendens. BIM is B. embellus. BSI is B. cymerolentalis. BMA is B. macanensis. BSM is B. smaragina. And BSG is the B. smaragina guitar. Outside Inside, I'm working on Making You Beautiful, Nick. It's coming along pretty awesome. Hey, I'm excited to see that. I'd love to be beautiful. I love it. Work your magic. It's been pretty cool to see the process. So when the final thing's out, I'm gonna be that's gonna be pretty crazy. I'm not gonna lie. C said, I don't know what they've done to these fish, but they've turned them into tumor factories. I wonder if they'll mention that. Oh, did I put the let me put the link to this in the chat if I didn't already. No, I did. Never mind. Diana said, mine are good so far. Would have bought them in aquarium co-op, but they only carry males. Ah, how dare they. We collected 14 breeds of Betta fish differing in tail type, coloration, sex, and body size. They performed whole genome resequencing of 727 individuals, resulting in 2.7 terabytes of clean sequencing data with depths ranging from 3x to 34x. To elucidate the population history of the Betta splendens complex and the origins of the domesticated Betta fish, we further sequenced 59 individuals from 6 wild species of Betta splendens complex with an average depth of 24x to co-analyze with 20 randomly picked individuals from each breed a maximum likelihood phylogeny from concatenated sequences representing the average genomic coalescent tree showed that the domesticated breeds form a monophyletic group relative to other wild species. Outside in said, my Betta is growing a lot of tumors. It's sad. Oh my gosh, Steve was right. In addition, consistently, in principal components, they form a close cluster distinct from other wild individuals. These observations are compatible with the hypothesis that all current breeds of the Betta fishes were domesticated from the same group of wild Betta splendens. This big Steve said co-op be tripping. The Betta cymerolentalis individuals form a monophyletic clade within the cluster of Betta embellus individuals in the tree. The lineages of Betta smaragina guitar are interspersed with B. smaragina and B. stictos, which form a monophyletic clade within this cluster and the average genomic tree. All right. Several clades of the fighter breed fall as outgroups to the rest of the domesticated breeds in the phylogenetic tree, which is compatible with the breeding record that the fighter breed represents an early domesticated form and that the earliest domesticated Betta fish were in fact breeds selected for fighting. Other breeds falling towards the root of the tree include veiltail and crowntail. However, these observations can be attributable to introgressions from the wild species and the fighter, veiltail, and crowntail breeds, as extensive gene flow signals are observed among different fish groups. Population structures, as revealed by phylogeny and admixture analysis, suggest that breeds defined by coloration or morphology are generally clustered together, although we note that this conclusion might be affected by the sampling strategy used here. The half-moon plaquette breeds, which are short fin types, distinct from the fighter and wild Betta splendids by a rounded tail shape and body shape, also cluster on the base of appearance, mostly related to color. The red, yellows, orange, turquoise, green, royal blue, steel blue groups of breeds carry substantial group-specific genetic drift that is noticeable in the PCA. All right. Regulatory variants in DMRT1 mediate the male heterogamity of B. splendens. To map the SD locus, we performed association mapping on all 727 individuals, consisting of 590 males and 137 females, using a mixed linear model implemented in Gemma. The kinship matrix and the first three principal components were used. All right. Let me get to the thing that I can actually understand. Here we go. Colors and color patterning in the betta fish. This is what I was trying to get to. Crossing royal blue male and female individuals produces turquoise green. Royal blue and steel blue offspring at the Mendelian ratio of 1 to 2 to 1. A previous study comparing the composition of pigment cells and scales among these breeds showed that steel blue differed from the others by lacking erythophores on the lower layer of scales. So let's go back to crossing royal blue and turquoise green. So this royal blue is S4E. S4E. Is that up here? Is that another one? My veiled tails are gaining fin rot, just the tip of the fins? Oh no. A GWA analysis coding steel blue, royal blue, and turquoise green identified a single locus at 8.9 to 9.19 MB on chromosome 24, and GWAS with the other coding schemes consistently mapped to the same location. Genotype analysis at peak SMP showed that turquoise green and steel blue are homozygous for different alleles, and 27 of the 28 the royal blue individuals are heterozygous, which is highly consistent with the codominant inheritance pattern. The copper breed of betta fish has a characteristic metallic appearance on scales, and breeding records indicate that it's derived from introducing the metallic gene into the genetic background of the steel blue breed. We performed a GWAS using copper as cases and steel blue as control, and identified two most significant peaks. The strongest one overlaps with the locus underlying the royal blue, steel blue, and the turquoise green color variant, and the other is located at the 10.37 MB of chromosome 5, with all peak variants residing in the SRGAP3 gene, a cytoskeleton regulator. Genotype analysis at the lead of the SMP of this locus shows that steel blue is homozygous for the T allele, while copy individuals carry one or two copies of the alternative allele C, consistent with the known inheritance pattern. Therefore, we consider the associated region a strong candidate for the hypothesized Mendelian metallic gene. Bob said, after my last betta got a tumor, I said no more domestic bettas. Entelos, erythophores, and xanthophores produce red and yellow pigments, respectively. An orange color can be conferred by either a pteridine component or a mixture of red and yellow pigment. The case control GWAS contrasting the orange breed with the red and yellow breeds identified a major locus. All right. Let me look at this figure real quick. So this shows the genome-wide association studies. Here we see the E. We see the red, orange, and yellow phenotypes. Okay. What do you guys like the most out of this? The royal blue or the turquoise? I don't even know. I don't even know what color betta I like the most. They call this mosaic. I want to see what they say about the mosaic. The mosaic color pattern is a spectacular phenotype in the Siamese fighting fish with multiple commercial names, including koi, candy, galaxy, lemon, and marble. Although the mosaic color pattern has been observed in koi carp and medaka, the underlying molecular basis has not been investigated. Here we perform GWAS with a case control design and identified 9 associated loci on 8 chromosomes, suggesting a polygenic basis underlying this phenotype. Ooh. The Siamese fish also show great diversity in their eye colors and at least 6 color categories, including black, white, yellowish, is that the scientific term? Yellowish brown, yellow, light blue, and brown. However, GWA mapping on each group did not reveal a strongly associated loci. Okay. Let's see what they say about some of these other phenotypes. Veil tail, crown tail, and half moon. Within the long fin breeds, there's much additional phenotypic variation, with veil tail, crown tail, and half moon breeds as some of the most distinctive breeds. Veil tail is the first long fin breed recorded, while later selection for a full 180-degree spread in the caudal fin ray gave rise to the half moon breed. The crown tail breed differs from the half moon breed by having reduced webbing tissue between the fin rays. The Dumbo phenotype. The pectoral fin and telos is homologous to the anterior appendages in amphibians, reptiles, and mammals. One breed, the Dumbo, is characterized by the overgrowth of its paired pectoral fins with more and elongated fins. They investigated the same phenotype. Okay. This is okay. What else they say? Oh my gosh, guys! I've been waking up so early. I'm not even used to trying to be awake at this time of day anymore. I'm not gonna lie. I'm not gonna lie. I'm fading a little bit. I'm just fading a little bit. Holy. I did link this in the chat, right? The great thing, though, is I put this in here. I put this in the Discord. So if you guys want to take the time to go back and actually, figure it out, please do. And leave a comment. Because I think I'm going to need some time to digest this one. Holy. Look at this. They got the test fish and the opponent fish. What the what's going on, man? I'm just scrolling through. It's like science. I don't know what I'm seeing. And then I just see test fish and opponent fish. Ichthyo Dude said, in the flesh, could be the result of lower blood flow to the outer edge of the fin. So interesting. Diana said, apparently aquarium co-op had too many people buying females for beta sororities failing and then returning them, blaming aquarium co-op, so they quit carrying females. What do you mean return? How do you return a fish? That's the thing? Okay, let's read this. This diagram seems pretty interesting with the fighters and non-fighters. So they're looking at aggressiveness and B splendens. So GWAS of fighter versus non-fighter individuals. The genes tagged by lead SNPs and peak are shown, and neural system related genes are highlighted in bold. 2 representative morphs of fighter breeds are shown in set. Experimental setup to quantify the aggressiveness of B splendens individuals. Aggressive indices of test fishes are recorded in the presence of opponent fish. Box plots of 8 aggressive phenotypes in fighter versus non-fighter individuals. Manhattan plots of GWAS use the charge score and aggression index respectively. Big Steve said, how is Ultra Premium Betta Machinensis doing? I think they're doing well. Actually, I need to split some of them up because there is a giant bubble nest and I have them in a tin. So I have a feeling it's about to get crowded if I don't split them up into places. So I think I'm going to pair them off. And I got a 5-gallon I'm about to open up. But they're doing well. I think I ended up with 4 males and 2 females. I think. Okay. I'm not sure. It seems like one of them I can't really tell. Okay. So we got this opponent fish. We got a camera. They're in a tank. They got a little bit of a separation thing going on here. And they have a test fish and the opponent fish. What's up, Mountain Greenery? How's it going? What's up, a couple of fish? Okay, so here they're testing different. They're plotting different behaviors. So we have mouth open, gill flare, fin flare, retreat, aggression index, times of air breathing, charge, and approach. Big Steve says it's the customer's fault he doesn't sell females. LOL. Alright. By synthesizing all 10 behaviors in an aggression index. So, yeah, they put all the behaviors in an aggression index. They identified a cluster of associated variants on chromosome 8. Okay. Let me see what they said about this aggression. Aggressive behavior is a complex phenotype involving genetics, endocrinology, neurophysiology, and metabolism. Male winners of Siamese fighting fish in 1-versus-1 fights have been selected by breeders for improved combating performance, thus producing the fighter breed with stronger aggressiveness, longer fighting duration, and more active in motion. We performed a GWS using fighters as cases and other breeds, or as controls, and identified 36 association peaks distributed across 21 chromosomes, suggesting a polygenic basis for the behavioral differences between the fighter and other breeds. The strongest association signal is found on chromosome 4 in the lead S&P and is located in the vicinity of 2 copies of ESYT2, a promising candidate for the aggression phenotype, as it is shown to promote neurotransmission and synaptic growth in Drosophila. We also highlight 6 other neural system-related genes that were tagged by associated peaks, including APBB2, that encodes B amyloid, a 4 precursor protein binding family, okay. And B member 2 protein is associated with neurodegeneration and Alzheimer's disease. Oh, fantastic. Fantastic. To study these behaviors, we phenotyped 10 different behaviors displayed during simulative fighting, including approach, charging, air breathing, gill flare, fin flare, jerk mouth open, pacing, retreat, and shimmer, and performed association mapping to identify genetic architecture for each behavior. A GWAS for charging behavior during fighting identified a strong associated SMP located close to 2 copies of GFRA2, which plays a key role in the control of neuron survival and differentiation. Notably, the association signals reproduced in GWS for the mouth opening behavior, suggesting a common genetic basic for these 2 correlated behaviors during fighting. Wow. Alright. I think that's what I got out of that one. Holy. I got another better one. This one's even more devious. This is the CRISPR one. Genetic manipulation of betta fish. Can we just get some glowfish going on? I mean, seriously. Yo, let's get the glowfish up in here. Team glowfish. I feel like that's got to be one of the most ultimate rage bait things about, you know. Just put a glowfish on the screen. That's why I should get some glowfish just to put on the on the cam. I'll probably get people coming to the stream just to hate on the glowfish. Get that abomination off the screen. All right, I put the link to this in this next paper in the chat, and I'm putting in the Discord as well. Holy, I don't even know how to read. What's going on? Okay. Genetic manipulation of betta fish. Oh my goodness. We got the Zuckerman Mind Brain Behavior Institute, Columbia University, New York, NY, Department of Ecology, Evolution, and Environmental Biology. Oh yeah, we got some people up in here. Betta splendens, also known as Siamese fighting fish or betta, is a freshwater species known for its astonishing morphological diversity and extreme aggressive behavior. Oh my god, bro. Oh my god, this stereotyping by fish, man. Not all bettas. Despite recent advances in our understanding of genetics and neurobiology of betta, your eyes are closed? My eyes are not closed. What? The lack of tools to manipulate their genome has hindered progress at a functional and mechanistic level. Smart Person says Mind Brain Behavior Institute at Columbia is cool. We're about to find out. In this study, we outlined the use of 3 genetic manipulation technologies, which we have optimized for use in betta: CRISPR/Cas9 mediated knockout, CRISPR/Cas9 mediated knock-in, and TOL2 mediated transgenesis. We knocked out 3 genes: ALK-AL2L, BCO1L, and MITFA, and analyzed their effects on viability and pigmentation. Furthermore, we knocked in a fluorescent protein into the MITFA locus, a proof of principle experiment of this powerful technology in betta. Finally, we used TOL2 mediated transgenesis to create fish with ubiquitous expression of GFP, and then developed the bicistronic plasmid with heart-specific expression of red fluorescent protein to serve as a visible marker of successful transgenesis. Let's go. Uncle Joe's Fish said, "So good evening, everyone." What's up, Uncle Joe? So, oh my god, bro, we got more. We got all the charts. We got all the data on the screen. Although this is some cool close-up photos of the betta scales and pigmentation. That's pretty cool. Wow. So this shows the CRISPR/Cas9 mediated knockout in betta. Yep. Let's see. This is E. E is representative images of betta individuals according to the ALKL2L genotype, which has microscopic images of the side of the body showing the scales and a iridophore cover. The color of the microscopic images appear different from the whole body photographs on the left due to the use of bright incident light under the microscope. In D, box plots show proportion of blue left and red right pixels in ALKL2L F3 crispants. Oh my gosh. According to the genotype. So they appear with A. We have a blue veil. They start with a blue veil x blue veil. And if you understand this stuff, you can see they kind of have plot out the whole thing. So we have Cas9 over here, Cas9 protein plus gRNA. They micro-injected into a zygote. Then we got the blue veil with the PO crispant. So I guess the Ichthyo dude said, "I don't think big box breeders really practice culling." Probably not. Minato says, "Genetics live stream. I clicked so fast once I opened YouTube." Yo, let's go. Someone's excited. I'm over here like just struggling to read. Holy. So okay, then we get they got the crispant, the CRISPR produced betta. They put that back with the blue veil. Then they got a F1 of the blue veil versus the crispant. They put that F1 with the blue veil. They put 2 F2s together. And then, yeah, we got some F3s. And you see we got plus plus. 1 F3's plus plus, which is shown here, what it looks like. I'm not gonna lie, this betta kind of looks like shit. Sorry. What's up with this? Its tail, it's all beat. Its fins all beat up. How are they keeping these things, man? Maybe these are like post-mortem photos or something, but my goodness. Uncle Joe says, anyone mess with wild bettas? Of course. Of course. I have a couple, but mostly Betta rubra and Betta alba marginata. Icto Dude said, my friend works with zebra Daniel crispins for a lab. They get their parent stock from the wild and don't use progeny to keep the line going. Oh, interesting. Huh. Interesting. So here the F3++, we got this guy. Then F3+-, we got this guy. But the F3-, this Betta does look kind of cool. It's like all black. I like the little spots in the dorsal. And it also looks the least beat up out of the three. Okay, so here we have a close-up of the scales. And then we have some saturation, some hue information. Minato said, Ithio Dude, Big Box doesn't do the culling. It's pretty much their suppliers giving Big Box their low-grade specimens. Yep. Diana says the scale close-up looks like peacock feathers. Yeah, it does. Speaking of zebra Danios, establishing genetic tools in Betta will advance their use as powerful experimental system to study development processes and behavioral traits. Genetic manipulation of Betta is facilitated by their reproductive biology. Betta fertilize externally and produce clutches of about around 250 eggs, each with a relatively large diameter of 1 millimeter. This enables the micro-injection of zygotes in a similar manner to methods for genetic manipulation of zebrafish and medaka. Nevertheless, asynchronous egg fertilization resulting from a protracted mating process can last many hours, coupled with a short interval between fertilization and cell division, as well as thick chorion, constituent significant challenges for genetic manipulation. Big Steve says he has Betta machinensis, Betta rubra, Betta smigrina, Betta alba marginata, Betta antuta, and Betta macrostoma. Big Steve with the big Betta flex. Holy. Although a few studies have successfully utilized these tools, their reported success rate have been low and none have reported germline transmission. Furthermore, although plasmid DNA microinjection has been used to make transgenic commercial Betta, the glowfish Betta, the more controlled and efficient TOL2-based approach has not been performed. TOL2-mediated transgenesis has advantage over simple plasmid injections, such as an improved rate of germline transmission and higher frequency of single copy integration. In this study, we established effective protocols for 3 different genetic manipulation technologies in Betta: CRISPR/Cas9 knockout, CRISPR/Cas9 knock-in, and TOL2 transgenesis. We used CRISPR/Cas9 to knock out 3 genes, ALK and LTK, ligand 2-like, AL2L, beta-carotene oxygenase like 1, BCO1L, and melanocyte-inducing transcription factor A, MITFA, and provide a phenotypic analysis of pigmentation and viability of alkyl-2L and BCO1L germline crispins. We also report the first successful CRISPR/Cas9 mediated knock-in, as well as the use of TOL2-based transgenesis in Betta. So, results. Steve said, I don't know what to do with these Antuta. I'm scared to change anything, but I'm scared the female will mess with him if he freaks out when trying to catch her. Ithio Dude says, weird question, but does anyone know what volume tank does Teen use for his fry grow-out racks? Looks like 10G to me, but not sure. I'm sure someone in the chat knows off the top of their head. I don't know off the top of my head, though. I feel like he uses. I feel like it's like a 20. I feel like I've seen him with the 20 long, and he's got a bunch of trays all across it. I think he uses a few different sizes. I don't know. I'll have to look up I'll look up the video at some point in the stream if you remind me. Okay. They chose Alcal2L and BCO1L as targets for CRISPR/Cas9 editing based on previous genetic mapping. The identified variation in these genes as likely contributing to blue or red coloration in the ornamental betta. Alcal2L alleles present in bluefish are associated with an increase in the proportion of blue coloration covering the body and a concomitant decrease in the proportion of red, whereas BCO1L alleles likely modulate red hue. JP Jeffrey says it's a 20 long. That's what I remembered. Diane said I could have asked him at the club meeting last Saturday. Parna Haiba Nanos saying hi to Mini Minato. What's up? What's up? How you doing? Although molecular mechanisms responsible for this phenotypic variation have not been studied in betta, Alcal2L is necessary in zebrafish for the differentiation of iridophores that contribute to blue iridescence. And BCO1L cleaves B-carotene and a red-orange molecule into 2 molecules of all-trans retinol. As a positive control of the induction of CRISPR/Cas9 mediated deletions, we also targeted a third gene, MITFA, using a guide RNA that has been validated in betta. We injected 1 to 4 celled embryos with a ribonucleoprotein RNP injection mix consisting of Cas9 protein and guide RNA. Paranahaiba Nano says this is about color layers coming in late. It's about a few different. I think they're kind of describing what they're doing it for now, but the title of the paper is Genetic Manipulation in a Betta Fish. And the pinned comment in the chat should be the link if you want to read along, read ahead, or you're sick of me reading things incorrectly. You just turn off the stream and do it yourself. Although the molecular mechanisms for this phenotypic variation have not been studied in betta, yes, Alcal2L is necessary in zebrafish for differentiation of iridophores that contribute to blue iridescence, and BCO1L cleaves B-carotene, a red-orange molecule, into 2 molecules of all-trans retinol. As a positive control of this induction of CRISPR/Cas9 mediated deletions, we also targeted a third gene, MITFA, using a guide RNA that has been validated in betta. We injected 1 to 4 celled embryos with a ribonucleoprotein RNP injection mix consisting of Cas9 protein and guide RNA. We interrupted betta 1 to 2 hours after they began mating, as this maximizes the number of embryos at the 1 to 4 cell stage, and injected these embryos using a very sharp needle made with micropipette puller and beveled using scissors, which facilitates the penetration of the thick chorion. We then allowed surviving embryos to develop for 5 weeks when they are about 12 millimeters in length before extracting their DNA from a fin clip and genotyping them using a T7 endonuclease. 10 of the 14 targeted fish for which PCR amplification of a genomic region surrounding the cleavage site was successful showed that T7EI activity indicative of Cas9 cutting activity. The rate of CRISPR/Cas9 mediated mutagenesis was lower for BCO1L, only 9 out of 21, 43%, and lowest for MITFA, 4 out of 29, 14%. Yet at sufficiently high rates to be a practical approach. Paranahaiba says, oh, interesting. Like in a lab, I bred domestic and know some people really knowledgeable in their genes. I've never heard of that. I wonder if they can make albinos with this method. Yeah, this is the method they're testing. I think is kind of similar to how they do glowfish stuff. Genetic manipulation. Because beta MITFA knockout phenotypes have been reported previously, although only in the P0 generation, we focused on Alcal2L and BCO1L mutants for further study. We crossed PO crispins to wild-type beta, the same color and fin type as the parents of the PO animals, BCO1L crispins to red veiltail fish, and Alcal2L crispins to blue veiltail fish, and genotyped the F1 offspring using a T7EI assay. 1 out of 2 BCO1L and 2 out of 2 alcal2L PO crispants we crossed transmitted a mutant allele. The BCO1L PO crispant transmitted a mutant allele to 3%, 5 out of 149 of its F1 offspring, whereas the alcal2L PO crispants transmitted to 47% and 80%, so 8 out of 17 and 8 out of 10 of their F1 offspring. Is there good for strains of albino bettas, or like putting albino gene into other species of betta? Albino alien betta sounds interesting. I don't know that I've seen an albino betta, I'm gonna be honest. The varying levels likely to reflect the degree of mosaicism in the CRISPR/Cas9 induced mutations in the PO crispants. Next, we sequence the targeted loci of 15 T7EI plus F1 offspring, so 5 BCO1L and 8 alcal2L, to characterize the CRISPR-induced mutations. Sequence analysis revealed a variety of mutations in the F1 generations of them. We chose one F1 crispant per gene that carried frameshift mutations leading to an early stop codon. We then crossed each of these F1 crispants in a second outcross to wild-type betta to obtain F2 generations containing heterozygotes, genetically identical at the targeted loci. Finally, we crossed two F2 heterozygous sibling pairs to each other to obtain a set of F3 animals in which we could compare homozygous wild-type, heterozygous, and homozygous mutant animals within a clutch. For BCO1L, the mutation we chose was a T insertion that led to a frameshift coupled to a premature stop codon, 21 amino acids downstream of the indel. Outside in said, what do you say about cleavage? Holy. Parna Basis said, the only albino bettas are ocular albinos. There are no true albino bettas. They're white/golden aliens, which were first bred by a Brazilian, and then Frank's Betta stole the credit, LMAO, but they're not albinos. Ithiodu said, not bettas, but Alan Bias has a couple papers about guppy color genetics. I know we got see the guppy genetics stream. I think I'll be able to understand a bit better because there's some really nice resources on that. Some good videos, and then there's some good papers. I have the PDFs of some of the guppy color books too that are pretty nice. And yeah, Joe P has a good video about them. Ocular albino can only occur in marbles as far as I know and can't be bred first since the fish are blind. Oh man. Our PO crispant was a female, and her F1 offspring that sired the F2 generation was a male, demonstrating that the mutation can transmit both eggs and sperm. Sorry, I went ahead a bit. Oh, no, never mind, I'm good. And that inheriting two copies of the BCO1L is lethal. Image analysis of the color distribution and hues of the adult fish revealed no significant difference in the proportion of red and orange pixels or of the joint hue, saturation, and value. Minamano says, Alan Works holds very strong. A lot of people use his charts for breeding and seeing what genes their guppy has. Yep. I mean, I got the guppy color book. We could do that. We should do a whole stream on just that one though. That's kind of crazy. Guppy colors are both more complicated and easier. You can predict much easier what comes out, but there are more hidden genes. Yeah. There's just such good pictures of all the different morphs in some of those books. All right. Therefore, allelic variation of the BCO1L between red and blue ornamental betta is unlikely to be composed of null alleles, and future reciprocal homozygosity assays will evaluate the involvement in red-blue fish variation. Ichthyodude asks, what's ocular albino? I'm pretty sure that just means like in the eyes, like the red eyes, perhaps. Parner says, with betta morphs, unless it's a solid color or something like metallics, you're working mostly with phenotype. And says, yes, just the eyes. For alcal2L, we chose CRISPR-induced mutation consisting of two base pair insertion coupled with a nearby single nucleotide mismatch. This mutation led to a frame shift associated with a premature stop codon 18 amino acids downstream of the indel. Next, we analyzed the impact of alkyl-2L in the F3 generation containing the plus plus and plus minus and minus minus genotypes. We found a significant difference in the proportion of blue covering the body of alkyl-2L compared to the other genotypes, with minus minus animals having only 25% as much blue as the plus plus animals. We got another chart. Let's go. We got another chart. A shows the experimental scheme for generating knock-in beta. B shows the bright field top, fluorescence only middle, and merge images of wild type non-injected beta, and PO GFP plus beta at 24 HPF. So here we have a wild type with a wild type. Over here we have the 5 bioattenuated GFP donor DNA with 40 BP homology arms. Oh my God, look at all the science over here. They got all this science together. They took the science and they micro-ejected the zygote. Holy. 1 to 3 DPF screen for fluorescence. Here we go, the POs. We got 2 glowing ones. PO GFP crispants, injection 1. Had a 30%, 7 out of 23. Injection 2, 24 out of 85. Injection 3, 24 out of 91. Look at them glowing. Glow bettas. Minamoto said red ocular glow bettas. Yeah, come on. Ocular, they lose all color. When they color up, their eyes continue colorless. So they're just fully blind. Oh my God. That doesn't sound good. Probably not ethical. Glow bettas are ugly as hell. Didn't they discontinue them? Heterozygotes not different from wild type, indicating that the alkyl-2L mutation is recessive. The extent of blue iridescence in alkyl-2L minus was reduced in both the body and fins, yet the reduction was more prominent in the body, consistent with previous findings, showing that the alkyl-2L mRNA is present at 210x higher levels in the body compared to the fins. Consistent with the reduction of blue coloration, the proportion of red surface was 5x more in the homozygous mutants compared to the wild-type fish. Microscopic images of the skin surface revealed that the blue iridescence mediated by iridophores is almost entirely absent on the body of alkyl-2 animals, but mostly normal on the body of plus-minus mutants, suggesting that one functional alkyl-2L allele is sufficient for iridescence in bettas. To better visualize differences in coloration, we plotted the joint distributions of per pixel HSV. The joint distributions of plus plus and plus minus are similar, whereas minus minus is an outlier. For hue versus saturation in both plus plus and plus minus animals, the majority of pixels have a hue value of sorry, 1 second. Sorry. Q value 20 corresponding to blue. Alright, let me get to my place. Minamano said, to be honest, if I got into bettas, I definitely would push GFP genes into other species. True glow bettas are better, but we're seeing how traits work, especially inserted genes, Minamano said. Parabin says, the still existing glow bettas are just underwhelming. They do go against ethical standards and can't compete in most shows. Unless this fish is cellophane or has very low color, the only way you can see the glow is in the eyes. Oh, man. That sounds mid as hell. I'm not gonna lie. I didn't realize they would look that bad. Okay. Let's look at this larva. Wild type. This is a TOL2 transposase mRNA donor plasmid. Micro-inject, you know what's up. 1 to 3 DPF screen for fluorescence. Wild type. PO. Transgenic. F1s. 1 to 3 DPF for fluorescence. Ooh, we got some cool photos here of Let me read. So B, schematic of PAB1 ACTB plasmid, bright field image of 24 HPF injected embryo left. Fluorescent images of 24 HPF embryo right. Okay. These are pretty cool pictures. Look at the little cute betta fish. Betta fish baby. Experiment 626. Next, to make a bicistronic plasmid expressing both GFP ubiquitously as well as M. cherry in the heart to serve as an additional visible marker, we cloned the 268 BP region upstream of zebrafish CMLC2 in front of M. cherry into PAB1 to make PAB16. We injected PAB16 into 1 to 4 celled embryos using the same injection mix components as above. At 3 DPF, we observed both ubiquitous GFP expression as well as robust M. cherry expression in the heart of injected embryos. 36 out of 72 surviving embryos at 3 DPF, but not at 1 and 2 DPF, M. cherry expression in the heart was bright enough that it could be used to quickly screen for transgenesis. Together, these results indicate that TOL2 mediated transgenesis is a highly effective method for generating transgenics in betta. Our constructs also offer effective visual markers of transgenesis and constitute useful starting points for cloning future transgenes. Parnas says, missing breeding them, they're so cute when tiny, legit look like little balls with eyes. There are some people who make artificial albinos by exposing eggs to a specific light. It isn't genetic, but it'd be cool to see something like an albino metallic. Yeah, that sounds pretty crazy, I'm not gonna lie. So they established protocols for 3 genetic modification technologies. Okay. It's honestly interesting hearing about how the micro injection. Prior to the day of injection, we made micro injection needles and injection molds, and prepared working concentrations of components of micro injection mixes. To make micro injection needles, we used a micro pipette puller model P1000. From Sutter Instrument, Novato, California, to pull 4-inch long, thin wall filamented gas capillary tubes. Oh my gosh, I can't even speak. Into injection needles using custom settings: heat 480, pull 90, velocity 80, delay 120, pressure 230, and ramp 467. This is a compressor. This is like music production settings. We then used micro scissors. 15000-08 Fine Science Tools, Foster City, to cut the pulled needle at a 45-degree angle under a dissection microscope so as to bevel the needle to facilitate piercing of the thick chiron, however you say it. The diameter of the tip cutting is 8 micrometers. We then store the pulled needles in 150 millimeter petri dishes on mounting putty, extreme putty. Tom Bao Suane GA. Until use. We made injection molds to hold embryos for injection by dissolving 1G agarose in 50 milliliter E3 medium containing 0.0005% methylene blue, pouring the hot agarose solution into a petri dish. Then placing the dry injection mold into the solution when it reaches 55 degrees Celsius. Man, they really just go into every single detail how they did this. It's pretty cool. Minamoto said you should look into rice fish genetics. I have a rice fish paper loaded up. I don't know if you guys gonna be awake for that. You guys gonna be awake for another one? This shit's long as hell. We'd have to skim through and look at photos and stuff, cause oh my gosh. You know what I mean? Yeah, should we do it? It's a pretty cool one. All right, let me pull it up. And of course, I got this link in the in the chat pinned right now for what we just read, and I got the link in the Discord live stream discussion chat. So if you want to go back and find these to read later, I'm linking them as I go. get you a fish streamer that can do both. Okay, I'm trying to keep up with it. Let me get this freaking Medaka. Yeah, this is a good one. This is a good one. Hold up. Let me make sure I got the open source link. This is called Genomic Consequences of Domestication and Diversification of Body Coloration and Morphology in Ornamental Medaka Strains. And this is from January 2026, so it's also relatively recent, which I usually try and find as recent as possible stuff if I can. That shrimp in the background is pregnant as hell. Yeah, I have three gravid snow white Amano shrimp in there. One already released and got buried up again. I just wasn't ready to try and raise the babies when she did it the first time. Apparently, Medakas are illegal here in Brazil, but they're one of my dream fish. Oh my gosh. Illegal? That's them being illegal is kind of crazy. You just always think of illegal fish being like snakeheads and things like that. Okay. Amano racing is hard as hell. Apparently, I have some friends in the area doing it, and they're starting to do it in large numbers. And they keep telling me it's not that difficult. So I'm going to try and do it and follow their methods. And shout out to Zebra Aquatics. He's the last person I talked to that was like, he was breaking down how he did it. And I'm going to try. I just I wasn't ready when it came around the first time. Let me move the camera angle a little bit. I just want to show some different scenery. Alright, I'm getting distracted. Sorry. I'm getting distracted. Okay. Now that I'm back, I want to move this over here for a second because I was trying to show the RoboCop. That's why I was even bothering to move things around. I wanted to show the RoboCops. Alright, let's get back into this. Let me make sure this is still big enough to read. And there should be some freaking photos. And I got the link in the chat. Those are Parodicinklus Horaldi. I got a bunch I'm trying to... Yo, you're in Brazil. You should... Come on, man. You know what's up. Give me the secrets. No. I'm pretty sure the Horaldi, I think it's just going to be getting them to the right age kind of a thing. I'm pretty sure they're cave spawners based on how, or crevice spawners based on how some of them have been acting as they've gotten bigger. Because they're starting to do that pleco thing where they like kind of pick a crevice and you see them with their tail and they're kind of shimmering about. They didn't do that at first. Now a couple of the bigger ones, they kind of seem to pick the spot that they're guarding. They're native to my river system. I've been trying to breed them. Oh shit. Okay, come on, man. Let's go. We can do it. Medakas are illegal, but daisy rice fish aren't. Oh, that's kind of interesting. Okay. So, this paper, just a preview. I'm just scrolling down so you can see there are lots of pictures and stuff. So, oh my god, like, we can get through it. We're going to be fine. Okay. They spawn the same as Autocinklus as far as I know on the glass. Okay. Well, if they do, I'll find out. But, I don't know. I feel the RoboCops like being on the different stuff than them. You know what I mean? They seem to like I could see them doing the glass or under leaf. I've seen a report of a nanopatoma, basically the RoboCop, and it was underneath a leaf, like a Anubias leaf or something. But I don't know. The Horoldi seem to be posting up in these little crevices, so I'll have to see what they do. But I feel like they just got to get to a certain age or something like that, you know. Definitely a lot bigger than when I first got them. Okay. There's a lot of Medakas. Let's just get straight to the sauce. Do you know how to ID male and females? I got some really egged females but no males when the spawning season arrived. Ichthyodude said, anyone into microbarbs? I do like barbs. I really want to... J barbs and butterfly barbs are on my list of things I like to breed one day. No. By microbarb, I don't know if that's a specific barb you're referring to or just smaller barbs, but I like barbs in general, so... Alright. I'm going to kind of cruise over introduction a little bit just to see. Alright. Medaka, their popularity is going crazy. There's over 700 varieties. For example, regarding body color, wild Medaka has a dull light brown color, whereas ornamental Medaka strains exhibit diverse body colorations such as red, black, or silver. This is due to changes in the composition and pattern of 4 types of chromatophores: xanthophores, melanophores, lucifores, and iridophores. The bright colors of ornamental Medaka strains date back over 200 years to the Edo period, including a yellow and white Medaka strain. Most likely, many of these phenotypes resulted from spontaneous genetic mutations and were fixed by artificial breeding. Exploring the origins of domesticated animals helps us understand the evolutionary process shaping their genetic architecture. Identifying the genomic regions selected during domestication can reveal the genetic basis for traits that are beneficial in domestic environments, such as less aggressiveness and disease resistance. The ancestor of ornamental Medaka strains are believed to trace back to the Hemedaka strain, a strain with a yellow phenotype from the Edo period. However, the region where the Hemedaka originated and whether Medaka domestication occurred once or several times in different regions remains unclear. There are 2 geographically and genetically distinct wild Medaka populations. The northern population along the coast of the Japanese Sea of Honshu Island, named Oryzaius Sakizumi, and the southern population, Olatibus, and the other parts of Japan. These 2 groups diverged more than 4 million years ago. A recent population genomic study on Japanese wild Medaka has shown that within both populations, wild Medaka could be genetically differentiated based on their local geographic distribution. In China, Taiwan, and the west side of the Korean peninsula, Medaka has a wide genetic variety, being called Oryzaius siensis. Based on each previous research on the wild Medaka population in East Asia, investigating the origins of ornamental Medaka strains through a population genomics approach is a valuable research objective. Jacob said Medakas have only 3 alleles. Oh, you said into breeding microbarbs. Yeah, the only barbs I've bred so far are the African banded barbs. Barbus fasciatus. Barbus. Fasciolatus. I bred myself, like, now I have like 30 of them. I started with 10. And I took a bunch to all the clubs and stuff. So I have my like group, but I haven't bred them again since the first time. From what I gathered, they spawn, I think I'm talking about the Horoldi, better during the wet season, I think, regardless of where they are currently, so around October to May. Okay, that could be good. Alright. Whole genome sequencing of ornamental Medaka strains with diverse phenotypes. To characterize genomic variation in ornamental Medaka strains, we collected 86 ornamental strains with a wide range of body colorations and shapes. For example, the Yoki phenotype shows a wild-type body shape with a vibrant orange color. Another is a strain, Black Deme, which shows protrusion of the eyeballs from the skull and deep dark body coloration. We identified and characterized 34 phenotypes in ornamental Medaka strains. Although several are known phenotypes observed in laboratory Medaka mutants, many were unreported thus far. To characterize the genomic variation of these strains, we performed whole genome sequencing on 201 Medaka individuals, including 181 individuals from 86 ornamental strains and 20 individuals from 5 laboratory strains using Illumina short-read sequencing technology. A total of 2.7 terabytes of data was obtained, corresponding to an average depth of 18 times the genome per individual. Genome sequencing reads were aligned to reference genome assembly of the HDRR strain. Alright, let's look at some of these photos. What's up, She Savage Chicklets? How's it coming? There's this paper on ricefish hybrids. Hopefully you can copy and paste it. But it's not too common to see ricefish hybrids in the hobby besides Medakas X daisies, but that too I've never seen before. Wait, what do you want me to copy and paste? Let me know. Let me know. Oh, and I put the link to this pinned in the chat, and I'm putting it in the Discord. The Discord as well. 1 second. Yeah, the In My Tanks Discord, the live stream discussion channel. I'm putting this in there. Very few people are really too into hybrids, TBH. For Medaka, probably, yeah. But I mean, there's plenty of fish that are hybrids, but people either just don't know or they conveniently forget about it. Like a lot of, like, Betta fish is a good example. You know, some of the cichlids. Alright. Let's look at these. I'll be honest, there's a lot of ugly rice fish out there. Like some of the bodies just look so malformed, and the eyes that protrude outwards. All right, let me make this bigger. Sorry to the RoboCops. Oh man. I like the white silvery Medakas the most, I think. Oh, and speaking of hybrids, I guess Libear's got a lot of hybrids. Wild Betta keepers / most ethical domestic breeders also don't like hybrids a lot, TBAs. It's just a common misconception that they're healthier. Minamano said I put the link in the Discord, okay. Lame King Medakas are the nicest, alright. There's a lot of silver in here. I think the darker strains are the ones where things sometimes get shaky. But the cull rates on some of these is just that's why one Medaka, I feel like I need to have tubs to really get into them and for them to do properly. But two, I just don't feel like culling fish all day. Like when Captain's doing her Medaka project is like the amount of culling that she's putting into to make that strain any good. I'm just not willing to do it. I'm really not. So A shows, so figure A here, all these different pictures of the Medaka shows the different strains, all the different characteristics they're looking at. Over here, B is a neighbor joining tree based on the whole genome sequencing data of ornamental Japanese Medaka strains. Representative strains with the phenotypes of distinct body colorations, including Kohaku, Yokihi, Orochi, Mayuki, Panda, and Rame, are shown around the phylogenetic tree to illustrate their genetic relationships. So here we can see the different strains and their relationships. Culling is essential for most fish with domestic strains, TBH. Wild types are easier in that sense, LMAO. Yeah. But I mean, like, for instance, like you brought up Corys and stuff. It's like there's a lot of fish where, at least as far as coloration, you don't have to do that much culling. I'm sure there's some health stuff that if you look close enough, you could probably find a decent amount to take out. But, you know, stuff like that. Even like shrimp, I haven't found shrimp to be that bad as far as just keeping the general population decently colored. I like the shiny ones too. Minamano said, yeah, but the more tanks, the better. Some people are confined by spacing for projects. That's why they don't usually go for it. It's true. T-Relly, you got to get a rack so you can get your projects in order. Okay. So here's the 34 phenotypes observed in the 86 ornamental Medaka strains. Strap in, because I'm just going to run through them. So first category is body color. So we got the phenotype Byokihi, which is the feature is increased redness, orange of the body surface compared to yellow Medaka, enhanced xanthophores, and loss of normal melanophores. We have white body color, loss of both the normal xanthophores and melanophores from the body surface. Blue, blue, loss of normal xanthophores from the body. Yellow, loss of normal melanophores from the body surface. The body color of the traditional ornamental Medaka, Hemedaka. We have gold, brown or wild type like body color, brighter body color than the wild type, decreased blackness of the body surface compared with the wild type Medaka. We have Kurobushi, body color pattern with black spots. This phenotype includes Sansoku and Kuraka. We got Akabushi, body color pattern with red spots. This phenotype includes Sansoku and Kuhaku. We have YWKO, body color without melanophores. This phenotype includes yellow, white, or Kahaku body color. Black, increased blackness of the body surface. Wow, straightforward. With wild Medaka type Medaka, increased melanophores. Orochi, more increased blackness of the body surface than the black Medaka mutants, increased melanophores. Mayuki, ectopic expression of the iridophore on the dorsal side of the body, reduction of melanophores and xanthophores from the body surface. Rame, increased number of iridophore expressing silver colored scales. Panda, decreased iridophore levels throughout the body, including the iris and peritoneum. Aurora, mild iridophore depletion throughout the body. Depletion of the iridophore in the operculum allows the red gills to be visible from the outside. The shade of the scale changes and has various colors such as green, red, blue, and yellow. Paranabensis said, hey, dude, that female in the rock looks plenty big to breed, and I see some males in your group, too. Yeah, man, thank you for noticing. I do feel like, that's why I feel like I'm going to be able to breed them eventually. It's just a matter of time, because I think I got a mix. And there's some that are just, they're so visibly broader and bigger, I'm pretty sure they're females. Let me see if I can put the camera on them. I'm going to zoom out a bit just so I can try and see more of them at once. Like this one. I feel like this one here is probably one of my potential females based on how big and broad she is. Yeah, as far as I know, they're a matter of time. You got to catch them in the wet season and do tons of cold water changes. I have 3 females in a tank because there hasn't been a ton of collection for them. I see them maybe once a day. The nice thing about the Heraldi is they seem to be pretty food motivated. Like, they do come out for the food pretty quickly. What's up? Tainta Lumite Cobaltium 27110. How's it going? Hello. Alright, let me pull the paper back up, but I was just trying to zoom in. All the Heraldi are kind of nicely lining up on this rock, though. I'll go with that. I'll go with this for the frame for now. I need to scrape the glass. Close enough. All I can do is try, right? All I could do is try. All right. We are reading phenotypes for rice fish. Aurora, mild iridophore depletion through the body. Okay, I read that one. Tourmarin is decreased iridophore throughout the body without the iris and peritoneum. Almost complete loss of iridophores and scales. Nijikin, ectopic expression of the iridophore throughout the muscle. Kohaku, body surface is partially white with orange spots. Sansoku, body surface is partially white with orange and black spots. Kuraka, body surface is partly orange with some black spots formed by melanophores. Albino, due to the lack of melanophores throughout the body color is yellow and the pupil pink. Black rim, dense melanophores are formed around the scale. Melanophores are formed around the scales, appearing as a black mesh. Fukumaku, blue silver colored peritoneum. And then we have body shape, Durama, with a short body axis. Handurama, mildly short body axis, longer body length than that of Durama mutants and shorter than the wild type. We have Hikari. The dorsal side of the body becomes ventral. The dorsal fin is shaped like a pelvic fin, and the caudal fin is diamond shaped. Ectopic iridophore is formed on the back, and the dorsal side shines silver. Parents said on the Heraldi, 100% of female, tiny spots and small nostrils. I didn't know the tiny spots and small nostrils. Okay, yo, you're making me kind of hype, man, because I really want to breed these fish. That's why I got so many of them. They were expensive, but they're just so cool. The Robocops I have in there too. The Robocops have not been as... I definitely had some loss from my first group. Whereas with the Heraldi, I don't think I've lost any of the Heraldi ever. I've had them a while now. But the Robocops, I mean, my one LFS near me, they don't even order them in because they said that they're too sensitive. Ooh, Parabensis, a guide to IDing male female Heraldi, 100% certainty. Males will have, I'm literally screenshotting this just to say, males will have wide and big nostrils, like very noticeably larger than females. Circumstantial, males commonly have stubbier face and commonly have larger spots. Larger and sparser, and females have more frequent spots. Okay, that's very interesting. My guesses were just based on like how wide their bodies were, kind of like body shape in general. But those seem much more reliable. I'm not gonna lie. Thank you for that. Okay, more phenotypes for the Medaka. Fin morphology. We have swallow. Some fin rays are longer than the wild type. Fin membranes do not elongate. We have Hernaga. All 5 types of fin rays are longer than the wild type. Fin membranes elongate partially. We have long fin. 6 fins, except the caudal fin, are longer than the wild type. We have real long fin. Oh my gosh, bro. Real long fin? I thought they were going to come up with something a bit more creative. All fins wild than wild type. Not only the fin rays, but also the fin membranes are elongated. We have no dorsal fin. Dorsal fin loss. Hilarious. For eye morphology, we have demi, protrusion of the eyeballs from the skull due to the skeletal dysplasia of the head, but no enlargement of the eyeballs. Big eye, tinmi, enlarged eyeballs that do not protrude from the skull. Sorry, big eye. Big eye is enlarged eyeballs that do not protrude from the skull. Then we have tinmi, small pupils, small eyeballs in some cases. Then we have suhogan, enlargement of the interior segment of eyeballs. Oh my goodness, there's some messed up Medaka out there. I'm not gonna lie. There's some super yeah. Perna said things I learned. Medakas are super messed up morphologically. Yeah, some of these strains should be represented in this photo. It's not like, I don't think it's like high enough quality where I can really zoom in on stream. but you can see some of the eyeballs and the gills being red, and the short. This 9. This looks like one of the short-bodied ones with the curved spine. Yeah, 10 looks like another kind of short-body one. I think these are in order from what we just read a little bit, because that was kind of making sense. Oh, here we see some of the protruding eyeballs. Jesus. Anything with protruding eyeballs, I just find to be disgusting. Love my natives. If you have a chance to get some Aspidoras roshii, they're absolutely must have. Mine have been laying eggs daily for a week now, bro. You're the best kind of friend. I'm not gonna lie. Brazil low key's got the nicest fish right now. A lot of the stuff that I'm interested in is from over there. You might like. Well, actually, no, that's not from Brazil. Where is that from? The black era tetras, the Empicthes luzae. No, that is from Brazil. I have those. Let me see if I can get those on camera. You probably know what I'm talking about. These guys. I don't live in the Amazon, so no cool tetras with plenty of cool bottom dwellers and libellars. We can't own a lot of our fish, though. Man. That sucks. That does suck. I kind of want to get those on the screen, but I'm not by that tank right now. But that's a fish that I've just been keeping, but I really need to get to breeding them. I have a good group. They're pretty big now. I probably get stuff from them sooner than the Heroldi. I don't remember the scientific name, but Piata cata vermelha mostly. Okay, I'll have to Google that. Oh, those are emperors, right, or related? I'm not sure exactly, but they are Empicthes luzae, I think is the scientific name. They were recently described. Man, that fish you just typed looks big. Well, not like super big, but like it looks beefy. All right. Let me see what else I want to read in here, because I want to look at what you sent too. Oh my gosh. Here we go. Swallow. Damn, that looks like a sad rice fish. Those fins do not look good to me. Real longfin. I think I just like longfin. Real longfin is too much. Those fins don't look too good. At least not to me. But longfin like that seems fine. Whenever I go collecting, the tetras are always nipping at me. That's hilarious. That's so cool though. Okay, here we go. So here we see the eyes. So here's Dime. So here's the eyes sticking out. Then we have Sue Hogan. The eyes sticking out more. Then we have Big Eye. So the eyes aren't sticking out with Big Eye. They're just larger. They're just rounder. Also, yeah, that one was described near the Patano region, I believe, so a bit far away. Recently, the studies around here have been with the aspis. Minamano says Big Eye looks adorable. Parna says the Big Eye looks like a sardine. And then we have Tinmei. I think this is a super small eye. And then we have some fin morphology. So this is the wild type. We have Hikari. Dorsal and anal kind of look kind of same. We have Handurama. So we got node. Oh my gosh. Then we got the Durama. This thing just looks like it's pleading for death. Ugh. And unfortunately it looks really cute, but I need to care for this horrid thing way. Yeah. It's actually disgusting. All right, so here we go. This is for phenotypes related to body color. And again, this is linked if you want to read all the details and stuff, but I'm just showing some of the photos. So for body color, we got wild type, and here's the yellow, here's the white, here's the Aurora Rame. That kind of looks pretty nice, and I like that. And then the Mayuki. I also like the Mayuki is like, I need more Lame. I need super Lame. We have Kahaku, Yokihi, that's just super orange. Panda. I haven't seen the panda in person before. Sensoku. You're in the scientific fish keeping server, right? Yeah, I am. Definitely, you should tag me in some pics, man. The information about sexing the horology is super useful. We got Orochi and we got the black rim. We got close-ups of all these too. Very nice. So here you can see they actually got the melanophores, anaphores, iridophores, and leucophores with the plus and minus all laid out here. So if you're interested in seeing the breakdown, that's up in here. Oh my goodness. I need to learn more about genetics so I can understand what I'm even looking at. That's cool though. Medalka Yalkin. Dang, there's no cranked up melanophore and iridophore strains. This is sad. That's what I'm saying, man. You know, it's all looking, you know, it's cool. I like the different photos, but the reality is most of the stores around here, it's not like they have the supercharged strains, really. All right, let me get my screen share on Google Chrome so I can actually show. Let me see. So I can actually show what you sent. And we can look at some stuff. Shout out to you guys for hanging out here so late. And also, we're going through these damn papers, man. We gotta stay awake. Holy. Okay. I need an insanely shiny Medaka right now. Come on. Let's get it. I'm gonna put the link to this. I'm probably not gonna read this right now because I'm just like I'm losing my lucidity. But this is the paper I'm gonna put in the chat. Hybridization between marine Medaka or Oryzaius dancina and Javanese Medaka or Oryzaius javanicus. That is definitely pretty different. Introductions of hybrids and reciprocal hybrids were conducted, and backcross hybrids between O. dancina and male ODJ were produced for biological and cytogenic analysis. Look at this. Oh, man. Yo, trigger warning. What in the world? Man, look at these spines. If you can even call, I mean, they put the S in spine with this one. They put the S in spine. I'm not gonna lie. I've never seen such jacked up fish before. I don't know what it is with Medaka, man. Yo, don't do this hybrid again. Don't do this again. Science has gone too far. Oh man. I'm just like totally hypnotized by the horror. It looks like a fish trying to evolve into a worm. Oh, this just is not right. I'm so glad most of this paper isn't in English, so I can't even understand the horrors. That's the crazy thing. I can't even read it. I can't even read this paper. But you just scroll down and you see this photo. It's just like, oh my god. That's bad. now we're gonna see. We're gonna see if our friend can like show us sexing these Heroldi. Let me see if I can enjoy it. I need to cleanse my eyes of what I just saw. Okay, hold up. So I got 2 right here. What do you think? Same sex, different sex. It's a challenge. Live fish sexing challenge. Alright, he says right one male. I was gonna guess the left one was female. Based off of the body shape, but I could also go off of, you said the males have sparser amounts of spots, which would track. Because the left one seems to have more spots. Parna says need to see the left's nostrils. Okay, hold up. Angle top view is best. Let me get the camera up some. 1 second. I can get a bit more of a top view. It's slightly more from above. I can't get like way on top, but I raised the camera a little bit so it's looking down more on them. Yeah, we need the nostrils. That's the problem. Can't quite tell if you're find me a picture I took of a. Let me find a picture I took of an obvious male. Okay. Yo. I love all my tanks like this to have just aquatic isopods randomly everywhere. It makes me so happy. Like. I just got aquatic isopods all over the place. See? Look at this guy. There he goes. Run away, run away. Well, there's not really anything to run from. Oh, ooh, look at that guy. Hold up. Can I get... There's one off in the distance that looks great. Look at him. This is one of the ones that definitely posted up. Always in the crevice. They also really like the overhangs. There's one that always hangs underneath. It's actually there right now. It's just too dark and you can't see. You said you want aquatic isopods. They're fun to watch. Yeah, they're super fun. I mean, I'm a little bit biased, but I recommend. I mean, you probably can find them somewhere near you. You look outside, some different water. What other fish should I look at while we're here? Second, I'm changing my my camera orientation here a bit. I'm gonna move the tripod around. I'm not gonna lie, SVC had this idea to do the genetic stream. I hope you're happy. This was another 2 streams in a row where I feel like an idiot. All right. Let me get to the next thing real quick. Are aquatic isopods and scuds the same? No. No, they couldn't be more different. Scuds are a menace and isopods are angels. Scuds are hideous beasts, and isopods are beautiful citizens. Yep. It's true. I don't know what to tell you. Alright. Moving my camera. Scuds are fake shrimp. That's funny. That's funny. Scuds are, you know, scuds are monsters. That's what it is. These are the barbs that I bred. Just so you can see. I guess we'll just do some macro to end. It's so hard to move around. I need a terror deck, bro. Yes, I have a group of these. And then I bet you know what this is. Yeah. You probably have some of those somewhere around you, right? No? Chapey Jeffrey got a lollipop. Smart person says Scuds are Sonic. Aquatic isopods are Mario. Oh my goodness. This is the Agazizi Alacure, I believe. I believe that's what it is. It's kind of weird tank stocking wise, I'm not gonna lie. You should get some dwarf pikes. I do have some dwarf pikes. In fact, I have some growing up that I bred. Unfortunately, the dad jumped, so I haven't been able to get another batch, but I still have the mom. But the babies are just now, well, now they're big enough, I think they're starting to pair off. But that is Regani. Regani. Wallacea regani, I think is what they are. They were Crinichichilla regani tabahos previously. I've been having some mixed luck. Some stuff I'm doing, you know, better than ever on, and other stuff it's like, oh, I'm still struggling with this. Like, oh my god. If I had a bigger tank, I'd get a pair to be predators to my live bearers. I really like them. The one that I want to do long term is just I gotta get my another pair going, because I have some babies. I got maybe like 8 that are almost getting to the right size, but yeah. I'm trying to get my camera around my cord, just not long enough. Show. It's gonna be so hard for me to show them because they're just, my camera setup is not mobile enough for these live streams. I'm trying to look around to see what I can show. They're legit so beautiful. How is breeding them? Oh, the barbs? Tane said Scuds are quite fascinating to observe in their own right. I know, but I'm just biased against them. I don't know what to say. Breeding these is really easy. They scatter a bunch of eggs, so you could set up like a container with moss over it, and they'd scatter a bunch of eggs in it. I just set them in a tank for a while, and then took out the adults. Oh, the Wallacea. Those were easy. They just paired up. They're very aggressive when it comes to defending their babies. Really nice, really great parents. I was devastated when the dad jumped while I was out of town. Because he was so, if someone else walked up to the tank, he wouldn't let his babies come out. But if I walked up to the tank, he'd do a little signal and they'd come up because they know they were gonna get fed. I don't know how he jumped. And the crazy thing is, he jumped across from the rack to another, on top of the lid of a tank on another rack. And if he had managed to just flop over like 1 inch, he would have been back in the water. I wouldn't have cared if he just ate everything else in that tank. That would have been fine to me. If I go freehand with the camera, I'll try and show some cichlids down here. So this is a Nanochromis pyrrhulius. They had a shit ton of babies. Oh wait, 1 second. You can't even see what I'm showing. Oh, give me 1 second. Gotta get it turned back on. There we go. Look at my settings. Alright. So these guys just had a bunch of babies. And I'm trying to show you them. But there's a bunch of stuff that I gotta touch on my computer so I can fix the view. Alright, there we go. So yeah, Nanochromis pyrrhulius. Actually really pretty. I'm hoping that it looks like that might be the mom there. Oops, I accidentally hit my spherical aberration. But you can see quite a cloud of babies over there. And I didn't even feed them recently, and they're still all out and about. So, if you look down there, yep. There's a whole bunch of them. Quite literally. I think last time I counted, I think it was like at least 40. For whatever reason, since the babies got bigger, the parents have basically elected to just, they'll stay in the shadowy area. They'll let their kids go out and just watch from there. I don't really understand why. I'm like, don't you want to be out there with them? But hey, shout out Fish NJ for the $1.14. Appreciate you. Thank you very much. Glad you're still around, hanging out. Last time I collected Seraphae CS, they decided they didn't want to live anymore over the course of a leak. Oh my gosh. Have you kept a Cherasidium? Oh, the common name darter tetra. Oh my gosh. No, I have not. I've not caught them. They seem really cool. That'd be a cool one to breed. All right. What do I want to look at next? What fish? Just right now, the way the room is set up, there's so many things in my way to move around. Ah, there you go. Now you can see her a little bit better. Very pretty fish. So that's the mom. The dad, what I like about these guys is the dad looks, they both look equally good. Like the dad also has those colors and the striping and stuff. And that's what I like. Another thing I like about them is there hasn't been any of the mom chasing the dad away. That they seem to co-parent really well, which makes things a lot easier. He might be off with a totally different cloud of babies, but as you can see, they're all the way up here too, so there's quite a lot of them. They're all the way to the front and with a bigger cloud. Oh, that's the dad. Let's see, the dad looks the same, he's just a little bit bigger. Did you get the pair from Aquatic Clarity? The Pyrrhulius? I did originally get this pair from Aquatic Clarity, indeed. I got some live bearers. They're not really... What cool ones do I have? I don't have too many cool live bearers. They're all on the other side of the room for the most part, unfortunately. I got a couple guppies. I got some red swordtails. Got some porthole light bearers. But I'm not too much on light bearers. I do have Ginga sulphureus, but I didn't get them on purpose. In fact, I would love for someone to take them from me. Parana said every person should get Ginga rubra right now. Why is that? Okay, let me see. I also, since I don't have my wireless, wait, which one's that? Ginga sulphureus. How do I describe? I don't even know how to describe. Some guy, like, I was supposed to get them for someone else, but that person dropped off the face of the earth. So some guy, like, handed them to me, like, yeah, this is for so-and-so. I'm like, well, I haven't heard from so-and-so in, like, 6 months, but I guess I got these guppies now. Oh, my God. Bro, look at these freaking crayfish. I'm about to have... I want to get another batch of them, but they grow so fast. Like, they grow so fast. They're really nice at clearing tanks of snails, though. I'm not gonna lie. But look in the other tank. Look how big that shed is. So I'm growing them. I'm putting like 1 or 2 in each tank to grow them, and I'm gonna try and pair them up. I will say my stir-by have been looking good, but the problem is I can't catch that one tetra in there. So every time they lay eggs, the tetra eats them all. But my stir-by have been having a good time. I just need to actually catch that last tetra, and then we'll be doing all right. Oh my gosh. Okay, let me get the soundtrack on. This place is a mess. They're super nice imports. Imports are closed. I can't get them living vicariously through people that own them. Yeah, I don't have Ginga, the one that you said. I'm sorry. Okay. Holy. Let me get back to these guys real quick. Yeah, they're not looking too bad. Just looked up the one you have. They're beautiful. Another library fan. I don't know. Some of the images, they look pretty good. I guess some of mine do look like that, but I'm just not. I have like so many projects that I'm like haphazardly wasting time by not having the proper space set up for and things like that. That like, I just want to get rid of everything that I'm not super passionate about. And the libraries is definitely one of those things where it's like, okay, they may look good, but it's just not what I'm into right now. I'm really trying to get into some of these tetras and stuff that I have that I've just been keeping and growing out. Try to DIY a ginkgo rubra with a feral guppy and a random blonde female. DIY ginkgo rubra. In F2 of a feral X albino pairing. That sounds like that could be. I mean, it could work. It just might work. Yeah. One day I'll have a Terra deck or something, so my camera's wireless and I can just walk around with it, point it at all my tanks without having to have it plugged into this damn cord. But for right now, I'm limited. I had to like set up my whole computer and everything by the tanks I want to show. I made some progress though today because I finally got my water running through a sediment filter and a carbon block. So now I can just fill my tanks without using the chlorinator and stuff, which has been nice. Library genetics are fun to play with, especially since they breed so much and so easily. No, I definitely get that. I think I'm just not ready fully to do genetic experiments. Like you said, you gotta set up the space, you gotta pay attention. So I think that's the main thing. I think one day I'll probably have a library phase, but right now I'm mostly just trying to breed fish and keep them kind of the same. I think that's mostly why I'm just not ready for libraries and the work that they take. At least to do something cool with them. But I do like and appreciate you guys sharing all your projects. If you want a rare fish, get some Hisonotus iota, Prana says. Once you manage to breed the Parados. Ooh. Yeah, if I breed the Parado... Oh, yeah. So the fish he's talking about is that the green one. Yeah, this thing isn't around, and if it was, it probably cost like $3 billion. I mean, I've seen videos of this one. If I could get it, then I would be over the moon. But I'm sure, I mean, the Parada sinklets are already more expensive than I probably should have spent. Here's a picture of what he's talking about, I believe. It's like a green one. I mean, if I bred the Parada sinklets, then maybe I could justify buying them because it's like, hey, I bred these. Maybe I can breed these other ones. But do you think any of my autos are big enough? Or do I need to grow them out some more? That's the thing. How much of it... Because it's kind of been raining a lot here, I'm not going to lie. So how much of it is just getting them mature versus environmental conditions? I'm fortunate, Harald, I live literally down the block from me. I can get them for $6. They do look big enough. They can grow to 5 centimeters at most. Okay. But maybe I need to figure out which food is going to get them ready the best. You know what's crazy? What they like respond to the quickest? If I like crush some snails in their tank, like on the side of the glass, they love rasping on the crushed snails. It's kind of interesting. I've gotten them they've eaten white worms once or twice when I put them in there, but they're not very good at it. They're not very good at it. I do put worms in there for them to eat every now and then, but they don't seem that great at eating them. High protein like tuberflex and baby brine shrimp. All right, I'll have to try that. I'll have to try that more. I haven't hatched baby brine shrimp in a long time. I've been mostly just doing worms. I'm lazy. I'm so lazy. Alright, guys. I should probably get off, go to sleep. Yo, for the people that are like me and struggle with things that require a lot of thinking, I'm sorry for another stream of reading these papers that are kicking my ass. But look, I'm trying to push myself here, okay? Next stream we'll do something. Maybe we'll have everyone send in pictures of fish and rate fish. Maybe we'll do that. I do gotta make a quiz, so we might have to do a trivia night, do a quiz, test ourselves and all the stuff we've been reading on stream recently. But yeah, that's some possibilities. Pirate says a fun stream. Yeah, appreciate you stopping by. I'm guessing. Oh, I guess someone posted a link in the scientific Discord. Shout out to them. Thank you for posting it. Also, my apologies if you showed up and realized I'm ignorant about science. But we're getting there. We're making progress. The thing with them is they're bad with food laying on the bottom. They need to learn how to eat from the floor. I see. Yeah, isn't that funny how you have to teach some fish to eat? It's just crazy. But I'll check out what you send in the Discord. If you're not in the Discord, hop in. it's been getting a little bit more active in there. We're getting things bumping. So I appreciate you guys. A lot of really cool, a lot of very, very cool photos of tanks and stuff that people have been posting recently. So we definitely got to do some raiding coming up. But I'm going to catch some Z's, and hopefully I'll see you guys soon. My work schedule, why it's been hard for me to stream is my work schedule. I worked 8 or 9 days in a row, and it was like I was waking up at 4:30 a.m. and then working, and then I'd get home and I'd be pretty tired. And some days I worked from like 5 a.m. to 10. So it's been a challenging schedule, but things are calming down now. So I should be good to stream a bit more consistently. And Ada the cat is now telling me that it's time to get off stream because she's starting to attack me. Yes, Ada, why are you starting to attack? Ah! See? On camera. I caught her on camera. Tried to attack. Ah! Oh my god. Oh my god. She's attacking me. You see this? Ow, Ada, why? I know why. She wants me to pay attention. Yeah. Yeah, I know. All right. Thank you guys. Appreciate everyone. Dwarf Cyclidiot said W stream, W learning, W Ada the cat. Okay, W guys. I'm off. Good night. More lighthearted streams to come. We pushed ourselves enough. Some of my tanks. And I'm out. I'm absolutely out.